Um, Welcome to attend this very well-organized meeting, and also thank Ashok for help on this thematic workshop. So before that, I just give a brief introduction about the AI for Science initiative in Singapore. So, okay. Yes, this is we have go over that. It's about so what is the AI for science? So we have actually have some argument about this. So eventually we find it's basically it's a general effort to utilize AI technology to solve this scientific problem. So the goal is of this initiative is bring together this AI or scientific or computer science expert with a top research scientist to identify, characterize, and eventually solve this problem in basic science. So here we draw a very, this is just the overlap of AI and science. So this area is AI for science, but it's different from science of AI because science of AI is more focused on the AI method. And it's also different from the general AI for X. So that is applied AI. And so this is show why do we need AI for science? Because for many years or many centuries, the scientific study is or conventional paradigm for scientific discoveries, observation plus analysis. So that it can be too slow compared with the modern experimental data is really become increasing very fast. It's really cannot imagine that a traditional way can work for manage that big data set. So here is one example which I'm very familiar with. Is AlphaFold. Probably people hear about that. Is basically they are develop this deep learning model to train their protein structure prediction based on experimental data in the PDB library. So this is an end-to-end pipeline. And then, after the training, this is a really amazing result. Show that this is experimental, blue is the experimental structure, and the green is a predicted structure by AI. You can say it's almost identical. So actually more than 2% of the cases, their accuracy is access to the experimental resolution. So that is one of the really very big breakthrough in this field, structural biology, also probably in the general life science. So because of this very important change brought about by the AI. So there's many of these global efforts on this AI for science. And I just list a few of these very important and typical AI for science initiative in different countries. And based on the scale, I just listed here. So we put a question mark here. This is Singapore. What we can do. So that is the NRF is they plan to put effort and build this Singapore-based initiative for AI for science. So this is, of course, I just list this advanced and why we needed that. So this is really have a good opportunity to provide a unique national level platform to promote and facilitate the collaboration between the AI of science expert and the scientific research scientist. So this is the architecture of this NRF, what they try to, I participated in several of these meetings. They have a plan to put a substantial amount of this budget for support the three domains are related with AI for science. So there's a foundational research on AI. So that is named as a catalyst. That will be a base for AI of science, AI for science. And then they will apply also to the important advanced priority sector. Those are like a important problem, like in the precision medicine or health. So here, we have sector and the data. So this is one of the two important component of AI for science. So here I just show some of the example of this, I think it's advanced area in Singapore for AI for science application, like bioinformatics or RNA biology and material science and so this is like a drug discovery or healthcare. So this is the milestone about this AI for science initiative. And we already go over this scoping study and also have this survey of this scoping study. And then we're here now in this thematic workshops is here. So after that, we will have, actually there's some of this, our NRF staff is already on the trip to the US and Europe. So we will have so after this, we'll have a FRC study report. So that will need you guys input for this like white paper. Then we will have a AI for Science and NTCI conference. So that is what we shown here. That is in July 23 to 24. So that is actually the NTCI. The Nobel Turing Initiative Conference that has been there for a while. So they collaborate with the AI for Science Initiative in Singapore to call for that meeting. So we have a number of this really the global leader in AI for Science. They will join us. They already confirmed the meeting. So here we organized about 12 or 13 of this AI for Science thematic workshop. And today we are here. So we have probably 5 more of these conferences or workshops. So the major goal of this workshop is try to get this white paper. So we will have like a 3 to 5 pages of white paper coming from the discussion of our workshop. Basically, 4 questions to ask. One is, what are the big problems in this field, in the AI for science? And what is really the challenge and bottlenecks to solve? And what is the possible methodology? And the last one is a transformative opportunity and outcome. So next week, we will have a Science of AI workshop. That is for the AI expert, because they will listen to us about what are the problems we are asking so that they can help. So we'll get some feedback from this AI expert, and then we'll update our white paper. Eventually, we'll merge together. Probably Kita and I will work on this study report. Then we'll submit it to the NRF. So this is a white paper. In the probably in August, we will have a call for proposal, AI for science. And they will ask you, if you are interested in this, you will submit like concept paper with more creative idea and concrete the detail of the proposal. So that is the overall outline about this workshop and also the AI for Science initiative. So thank you for your attention.
Would it be a painful situation of actually running a few minutes at the core? I have the opportunity to actually ask for questions from the or Professor Yang. I think it's very important that we understand what is being proposed in the AI for Science initiative. And this is the opportunity for us to understand how the initiative is going to move forward, how the white paper is going to be delivered, leading through in September. Proposals, which I hope many of you sitting in the room all day. So are there questions for Chow?
That last slide was very interesting.
Could we have the last slide up? Yes, I can go.
Okay. So the Science of AI workshop. So I gather that would be kind of like a pure AI people. Is that the idea, or it will be domain?
Even this one, Science of AI workshop. This is meant for the AI expert. It's from like a computer science and from different institutions. So they are like try to address the question we are asking from AI for Science, because they are focused on purifying on the AI methodology. So they don't know what is the problem we want to ask to solve. So this is really the purpose for the AI for Science initiative, because we want to put, bring together of this AI expert and the scientific, the researchers.
Sounds great. Well, one more question. Concept papers. Those are essentially like rudimentary done for.
Yes. Yes. Yeah, we will have this, the call for proposal about the, the outline and the policy and the requirement. Okay.
Yes.
And, the white paper is only one white paper for one workshop. So usually it's the lead, like, Ashok will be in charge of this white paper. Yes.
Talk about all the other things. So the workshops are each going to be based on white papers to
I'm just curious, this one is RNA biology. So when we talk about RNA, what about DNA?
So perhaps I can ask that, because I'm organizing this workshop. So in fact, if you don't mind, may I perhaps answer the question through two provocative questions I have for Yang,
Okay.
and hopefully the answer will then come out. Yang, if you don't mind, I have two questions. I think we would all accept that AI as a science is emerging as a very powerful tool to do all sorts of things. But tools that can open new directions, I will make the provocative statement that they may be somewhat sterile in the absence of context. In other words, how AI is used for drug discovery may be very different from how AI is used to analyze single-cell RNA data, nodding to Shyam. So how are you and Kedar going to address this issue of setting the context for AI in science?
Yeah, this is a good question. So we are certainly very selective, because Singapore is there's limited populations, so we need to focus on this budget really on those important problem, and not only in science, also is urgent and important for Singapore. So we'll consider the also the existing expertise in Singapore. So one goal is try to enhance the leadership of existing expert, which is many of our expertise are already among the leader of this the global community. So what we try to enhance that. Second is some of this project which is important for Singapore, specifically like what you proposed in the AI for science, AI for RNA biology, and you have specifically you propose like a genome which is specific for Asian and Singaporean. So those kind of more localized projects is especially welcome.
Thank you, Yang.
Yeah.
I hope that answers your question, because essentially the way I envisioned this particular workshop is to use RNA biology and its therapeutic applications, an area in which Singapore has already decided to make investments to address this issue of how does one set AI into context. So you'll see today, actually, there are 3 themes. Themes number 1 and 2 refer to specific potential applications of AI in the RNA space, first in analyzing high-dimensional data. Second is a specific issue of an application in predicting macromolecular structure and function for drug discovery. And the third is perhaps the most open, yet as a medic, from my perspective, one of the more exciting arenas. How can we use AI to begin to move into the clinic? And we can only skim the surface of some of these themes, but that is really the motivation behind organizing the workshop and the themes in their form today. I hope that answers your question. Are there any other questions? Yes.
So in that scenario, presume is the one going to study report that how aligned it be to the medical jobs and all? Is that going on different?
May I take that, Yang?
Yes, please.
From my experience with RNA, where we spent 1 year preparing the sort of FRC report and then later moved that forward to an actual funding initiative, I would say that like any sponsor of research, what NRF is looking for are ideas that are articulated in a way that justifies investment on a large scale. So all of what we propose may not get past the finish line because it may be shot down on the grounds that it is not aligned, on the grounds that it is not justified, or of course, as any research, on the grounds that it is not feasible. So what we have now is an opportunity to feed in to the process, which I think is something perhaps new in the Singapore environment. And I think as a relative newcomer here is so important and necessary for researchers to be telling NRF, look, this is what we feel is important. Yes.
Thanks for sharing. So I was wondering what the timeline leading to this milestone. And right now I understand that at the workshop part, right?
Yes.
Do you know the industry in such a workshop, or is it just intrinsically gathering KOLs or people before the industry? And then my other question is there a role for antimicrobial resistance to like public health interest to partake in this AI initiative?
Yes, this is the timeline which we have put together. Yeah, this workshop we are here is about the end of April. And we have some of this, previously, just like when we decide different, the topic and the thematic workshop, we did, have some argument about how we put the healthcare, because we want to really focus on basic science of AI. But everybody, appreciate this importance of healthcare, and so only until the last minute we try to integrate the healthcare in different topic into some of the workshop, which I think we have. One is this in the AI for genomics, AI for genomics. And then here, actually, Ashok include some, the last section touch about this, the health care.
Meant to be an exemplar, not a sort of end or exhaustive discussion. So, yeah. So I think we will rapidly run out of time and become late. So thanks again to Jan.
Yeah, we still have, I think we still have some of this, discussion, even eventually we have some conclusion. So we have a lot of time to discuss.
That's the idea.
Yeah.
Thank you, Jan.
You're welcome.
So let me then begin to kick off the formal sessions. I do have a few things to say to participants in this meeting. First, of course, I'm Ashok Venkitaraman, in case you had not connected my name with what was on the slide. And I'm chairing and have helped to organize this workshop at Yang and Keda's request. I think I've given you earlier a sense of what it is that we're trying to accomplish, but let me try and set some of this into context. So the first piece of context is something that Yang has already spoken to, so I'll just remind you of it, which is AI is revolutionizing our daily lives, let alone the way that we approach science in many different fields. So it's something we cannot ignore. In Singapore, we have to stay abreast if we are to utilize AI for our lives and for our scientific progress. Second perspective is from the perspective of Singapore, and I think this deserves some thought on the part of all of you in the audience as the day goes forward. Singapore has many huge advantages. It is a relatively wealthy, well-developed society, specifically in the realm of infrastructure. As a newcomer, I can say that I've been very impressed with the level of investment, with the talent, and with the infrastructure for accomplishing many things across the island. That said, can a country of some 5.5 to 6 million really compete on the world stage? And if so, what is it that we should be aiming to do? So one obvious narrative is, how can we use science to improve our condition, health, commercial translation here on the island? But that behooves the question, do we do that by investing our resources purely into making things or into understanding things? This is obviously a debate. I would venture to suggest, from my experience working with pharma, biotech, not in AI, I hasten to add, but in drug discovery and medical application fields, that companies tend to go where people know things that they don't know about. It's very rare, unless you have a low unit cost economy, that they go to places where people can make things. And that's because unit costs and methods of production are changing all the time. And it's very difficult to stay at the forefront in that arena, whereas it is arguably possible to stay at the forefront of understanding things, giving new perspectives on things, and thereby attracting industry and also implementing a health impact. So what does that mean in the context of Singapore? I think personally that it means many things. First, we are a relatively small place. So to compete with the best in the world, we have to be careful in selecting what we do. We can't do and should not do everything. We have to ask, as Yang has alluded to, what can we do in a competitive way, in a differentiated way, and how? Second, because we're small, it behooves us not to create more and more silos. Competition is good, but we should be competing with the outside world more than between ourselves. And the major institutions involved in research, for example, the universities like NUS, NTU, agencies like A*STAR, really need to work together more effectively for us to be competitive. The last thing is, how do we measure progress? So far, for many good reasons, we have focused on measuring productivity. How much does somebody accomplish with research funding? But there's a missing element in that, which is impact. many of the world's most impactful scientific discoveries that lead to health and commercial impact cannot be measured and should not be measured in terms of productivity alone. So we need to rethink the way we evaluate and do and fund science if we are to be competitive. So in that light, this workshop, I have asked all of the speakers not to simply talk about their own work, but to address the questions that Yang posed at the outset. What is the progress in their respective domains that have led to the current state of the art? What do they see as the major challenges? And importantly, what do they feel that we can do in Singapore to work together to achieve differentiated peaks of excellence in our research in this area? As I said before, and I won't explain again, RNA is just one area in biosciences in healthcare, and we hope that it will serve as an exemplar in many other areas. Last but not least, please do participate from the audience. We need your ideas if we are to reflect your views accurately to the NRF in designing the white paper. So this phrase on the internet, which my children tell me about, lurking. Please don't lurk, participate. And with that, I'll hand over to Shyam Prabhakar from the Genome Institute, who's going to give our first talk.
Thanks very much, Ashok. Great to be here and really very thoughtfully designed workshop. So excited to participate in that and honored to be one among the first speakers. All right. I really took your message seriously. So I tried to think about what can Singapore do that's competitive, playing to our local strengths and, across the community. Where is it? So, okay, next slide. Right. So first, maybe a little bit of thinking, at least the way I see the landscape, right? Because AlphaFold and ChatGPT are to a large extent why the local community got excited. And before that, it was driverless cars. But what is it about? But these are more biological or close to biological, right, as a language model. So what is it that, what's the innate nature of these, the first class, right, is based on public training data. Everybody in the world has access to that data. So you're competing on having brute force compute power, plus some intelligence in how you do your model training, okay? That's one class of very powerful deep learning algorithms. The other class, this press release just came out yesterday. Some of you may have seen it. $1 billion funding for this company. David Baker, a protein structure guy, is one of the key people in this. But here they're also doing proprietary data generation. So that's a different model. So of course, he's going to use the protein structure prediction AlphaFold type methods, but also generating competitive advantage through having their own data generation. And I think for Singapore to compete, we will have to combine these two to have a competitive. We can't just say we'll have bigger compute than other people. That's hard. Next slide. Next slide, please. Okay, and so far, whenever you say AI, people think reflexively neural nets, but in my field, single cell that Ashok alluded to, it's quite clear that, okay, so here's SCVI, the most popular neural net algorithm in my field, supposedly the best algorithm in the world for creating embeddings for single cells. Each dot is a cell in transcriptome space, and there's a 2D representation of the embedding of those cells. Based on this autoencoder neural network. And then this is embedding based on traditional machine learning. And you can see very easily by eye, right? The embedding based on traditional machine learning actually looks much better. You can see a lot more structure and a lot more populations cleanly than the embedding based on the supposedly best algorithm in the world. Right? So this highlights 2 points. 2 points. One is neural nets are not necessarily the best solution for all data types. And the other is that actually it takes a lot of domain knowledge to even know that it's not the best solution, because the machine learning people in the field think that this is the best solution. They look at figures like this, and to be honest, they have no idea it's a bad outcome, right? So what are some of the reasons why you might not want to use neural nets? If it's a relatively small data set, even this is million cells, but it's still a relatively small data set, then you have overfitting instability. Then if the model needs to be explainable, like you want biological mechanism to design drugs, then you need an explainable model, right? It can't be a black box. Then excessive compute cost and time, which is always something to factor in. And of course, we're heating up the planet with all the GPUs. So all things to consider. So I'd say we're trying, at least for biology and RNA, we want to do a combination of neural nets and more traditional methods. Next slide. So, so driven by, okay, what's exciting in the field is there are so many new RNA data types. And so you need correspondingly new AI methods that analyze them individually and combine them intelligently. And so then what can AI do based on these cool new technologies? And then based on Singapore's strengths, how can Singapore be competitive? That's how I try to structure it. Next slide. Right. So I'll start with a very nice example from Melee. So Renalmo is an RNA LLM that Melee just created, just made training on a public database of RNA sequences, but with some intelligent AI. Next slide. So he's trying to predict RNA secondary structure, and you can see here very briefly, because it's all boldface, that Renalmo outperforms all of the state-of-the-art RNA secondary structure prediction algorithms on virtually almost all classes of RNAs, right? That's remarkable. So best in the world, right? Homegrown. So this is certainly something to aspire to. But what this tells us is that even with public data, we can be internationally competitive. And this RNA language model could be is a foundation model, could be a workhorse for all kinds of applications, which I'll come to later on. Next slide. And this is from Jonathan, who's also in the audience. And Nanopore, here I talked about cool technology, super cool technology, long read sequencing of RNAs. Plus, you get the covalent modifications of the RNAs, which have very distinct biological meaning. Plus, you get the genetic mutations also that are in the exons, right? So this is one, you can say unimodal data, but actually you can transform it into multimodal RNA data from the same data input. And Jonathan has a whole host of algorithms for processing, AI algorithms processing this data. And you can infer RNA splicing, cancer transcriptomes, and then RNA modifications, as I mentioned. Very exciting space, and I think these methods can be combined with others for basic science as well as translation. Next slide. All right, and then I want to go back to single cell again, single cell transcriptomics, almost boring now because the field is 15 years old, but now it's getting exciting for a very different reason. 2 studies to show you that it can actually give you something translatable. So here, working with Thiong, we showed that we can find biomarkers that at diagnosis can predict whether this chronic myeloid leukemia patient will or will not respond to imatinib, which is standard of care. And it's a very important decision. If you know that the patient won't respond, you want to quickly go to more aggressive drugs. You don't want to wait for the patient to fail the treatment. Okay, so we're using single cell transcriptomics alone. We predict some biomarkers. It's not in this paper, but Thiong has validated those using a FACS panel in an independent cohort. Again, retrospective. Now it's very exciting. We're looking forward to going to clinical trials for these markers. So this is where the rubber meets the road, right? This is a slightly upstream example where single cell transcriptomics, again, we found this very strong transcriptomic signature across cell types of type 1 diabetes. But this one person, this blue person, was supposedly healthy, but had a blazingly strong type 1 diabetes signature, and turned out that that person got type 1 diabetes 4 years later, which we did not know. We just went to the clinician and said, come on, there's something happening with this person. Sure enough, that person died. And this transcriptomic signature preceded the high GAD1 titer on any other known clinical markers of type 1 diabetes. So both of these tell us that single cell RNA can predict drug response, can predict future disease. And I think that's a great opportunity for precision medicine in Singapore, right? Next slide. All right. So coming to single-cell RNA-seq in Singapore, either Asian Immune Diversity Atlas with, again, some people in the room and on Zoom probably, Jay, John Chambers, Boxiang, who's speaking soon, Don Chong, world's most diverse single-cell transcriptomic atlas. And you can see all of the Asian populations that we have here from many countries. And we found remarkable things about Asian biology. For example, SG Chinese, SG Malay, SG Indian, eightfold difference in expression in multiple cell types between Chinese and Indians in Singapore for this UTS2 gene, which in the UK Biobank is very strongly associated with basal metabolic rate, body fat percentage, and so on. And this is like a GWAS hit. And that GWAS variant explains, maybe like a 1.5-fold change in expression. But between Chinese and Indian Singaporeans, there's an 8-fold difference in expression, right? So what might be the consequences for metabolic phenotypes in Singapore? So this is an example of the Asian biology we can get from doing this kind of thing. And so what's exciting is to do multimodal AI, combining with a massive data set of genetic data set that Precise is generating, genetics and EHR, add some RNA on top of it from moderate size cohort. In fact, this is a Precise SG100K cohort. And then maybe you can predict disease for a whole range of diseases. And you can understand why there are, precision medicine differences across population in Singapore. Next slide. All right, that single cell, moving to spatial. Single cell is like taking a car apart and looking at each component, right? You don't learn so much about the car. If you see the car, the pieces of the car assembled together, then you really see how the car functions. And that's the concept of spatial omics, okay? And this is, again, collaboration with a bunch of people across the country. Here's an example, an exciting example of what you can learn from spatial omics without deep learning, again, necessarily, although deep learning may, we are adding to this with deep learning. This is a colon tumor. These orange things are the cancer cells. You can see here these little tiny orange blobs are the cancer cells budding out from the main tumor and forming new tumorlets or maybe even metastasizing. These are the dangerous cells. And we see from the RNA that they have a different RNA biology. They have this blue RNA state, whereas the cells in the main body of the tumor have this pink RNA state. So what are the markers of this blue RNA state, and can we target them with drugs and potentially arrest tumor growth, tumor spread, tumor invasion, metastasis, and all that, right? So there's an example, and there's many more examples, many different ways in which spatial omics can give you druggable biomarkers that you could not have gotten with single-cell RNA-seq. We have been doing colon cancer single-cell RNA-seq for 10 years. We never knew about this. The first spatial omics project with primitive spatial omics technology and primitive data and analytics, okay, not primitive, but relatively, we found something incredible, right? So I think the future is very bright for this. Next slide. And so that's the inspiration for the Tissue Map program, which is, again, a very broad program across Singapore to find cell-cell signaling interactions using spatial omics for diverse cancers and non-cancer indications using multimodal AI. So basically, spatial omics data, digital pathology data, H&E, clinical data. So that's the trimodal data set. And you want to learn embeddings, consistent embeddings across all these three modes that can tell you the patient's state, and that can also tell you the cell-cell signaling interaction. This is an example of a cell-cell signaling interaction we found using spatial omics that we feel is potentially targetable, and it's actually active in, again, the proliferating cells at the edge of the tumor nest. Next. So I think Tissue Map is an example of proprietary data that we are generating in Singapore on a very large scale, on which we can train our own AI models and have a competitive advantage that way and find some biomarkers that other people can't find. We're not the only people in the world using this model. Other people are generating. So there's Okin is a startup with $150 million US funding that's doing a similar strategy. Our funding is not $150 million, but I think we have a unique cohort, and we're already well underway. This is an example of the multimodal AI you can do with this H&E plus spatial omics data, where already so Grace and Hui Kwan and Tian Tian from SARC are driving this GNN approach, just initial plain vanilla GNN approach is already better than state of the art at taking H&E data, which all clinicians have, and predicting spatial omics data. Next slide. So coming to the grand challenges, all these exciting technologies, all these exciting preliminary findings, what does that tell us about the things in which Singapore has some strength? And also I'm going to propose some ideas that are a bit out there, because it's about grand challenges, not just challenges, okay? So the first challenge I've already more or less discussed, AI for drug discovery. How do we combine AI with all kinds of cool RNA technologies on clinical samples to identify diagnostic and targetable biomarkers to stratify patients, which we're already doing with single cell data, but stratify them much better for treatment and define the drug mechanism of action, right? So that's the AI for DD RNA AI challenge. Now I'm getting more fancy, okay? I've shown that single-cell transcriptomics can predict disease risk in a very unexpected way, okay? But can we get even better data? And can we compete with polygenic risk scores, which is really the state of the art for predicting disease risk, right? That's what, when you say precision medicine and predicting disease risk, it's about the genetics. But we're in AI for RNA session, and RNA, I believe, based on our results and based on other people's results as well, extremely promising for predicting disease risk. So I'm going to split that into, and it's not just RNA, right? So RNA plus electronic health records, plus maybe add genetics and so on, multimodal methods. For genetics, you need to sequence 100,000 Singaporeans and then 450,000 Singaporeans and so on to get statistical power. RNA has stronger effect sizes. You can get statistical power for discovery from a 40 versus 40 cohort. That was a T1D cohort, right? So you don't have to do RNA omics on all 100,000 people in SG100K, for example, can be a subset. That's the powerful thing. Now, here we're getting even more fancy. So here it's just combining RNA to polygenic risk scores, which is about germline mutations. Can we combine RNA with somatic DNA mutations? And of course, the exonic somatic mutations will be visible in the RNA. So combine that with the transcriptomic states to predict what's happening in aging, which is replete with all these somatic variants, right? As you grow older, you get all these DNA mutations, but they express themselves through the RNA and then through the proteins. So can we use these two together to say, okay, we predict that, for example, clonal hematopoiesis increases your risk of coronary artery disease, increases your risk of high blood pressure, all kinds of inflammatory conditions, and of course, leukemias, right? Cancer. So can we predict all these phenotypes in our aging population and say, okay, you are at higher risk. Let's prioritize you for enhanced screening, maybe some preventive for metabolic conditions, some preventive interventions, and then early diagnosis and so on, which is really important, right? So that's how I think this can be useful. And finally, this is a term Millet came up with, generative RNA biology. Can we take these language models and come up with designer RNAs? It's not a crazy idea. People already have been doing designer proteins for a long time. So designer RNAs with all kinds of properties, I think this is within our grasp right now. Thank you.
Thank you, Shyam, for really what was a perfect kickoff. As you know, Shyam addressed all of the issues I challenged the students with. We're going to have a long discussion session after all of this. So please save up your questions, dinner or up next. And I will be very aggressive in enforcing the 15 minutes.
Good morning, everyone. So speaking. Okay, thank you. Yes, today I would introduce the single-cell foundation models for drug discovery. Thanks, Zhang, for the very comprehensive overview of AI and RNA. So, and then I'll focus on this single cell, which is a little bit like old-fashioned technology. But with this technology, we are entering the big data era, and how we use the large foundation models to dissect this big data to derive biological insights. So with this AI and the big single cell data, we like to address several major challenges in current cancer treatments. The first challenge is often the off-target toxicity, because in the old times, most of the drug targets were derived from bulk analysis, which ignore the intra-tumor heterogeneity. Within the tumor mass, not all cells are malignant. So people are using single cell or mixed to pinpoint to the malignant cells to identify a better drug targets that shows a minimum of tumor expressions. And second challenge is the highly variable efficacy among patients, which is due to the patient-to-patient heterogeneity. But with single cell analysis, which is relatively still very costly, so within one study, the sample size is relatively small, insufficient to capture the patient-to-patient heterogeneity. Therefore, researchers has come up with these data integration strategies to scale up the sample size to characterize the patient-to-patient heterogeneity. And the third challenge is often the therapeutic resistance, and which may come from these complex cell-to-cell interactions or within cell molecule-to-molecule interactions within the tumor microenvironment. And all these interactions are very much understudied because of the limitation of existing technologies. Therefore, scientists have adopted the latest spatial or mixed to dissect these intra and intercellular interactions and try to identify alternative mechanisms of drug resistance. So, and as we have observed in recent years, single-cell assay has been adopted to for drug discovery and development. For example, it can be used to identify drug targets and to select the preclinical models to monitor the drug response and the disease progressions to justify the patients or subtype the disease. And then this shows a very conventional single-cell analysis pipeline for biomarker discovery and patient stratification, as we have learned from Shyam with the leukemia study as well as the diabetes. We recruit the patients and then collect the specimens from the tissues or liquid biopsy, and then we sequence a single cell sequence the samples, and then we perform a canonical bioinformatics analysis, statistical analysis to identify the cell type or cell state differentially abundant in responder versus non-responder, or genes that are upregulated or downregulated in responder and not responder. So this approach is like relatively a small scale and with existing canonical statistical tests or like traditional conventional machine learning methods. But with the advancement of single-cell technologies and the reduced cost in this technology, we have observed an exponential growth in the single-cell study in terms of both the number of samples and the number of studies. So with this increase, we have accumulated. This field has accumulated massive numbers of data in the public domain. So whether we should make use of this public domain data first before generating in-house data and to mine these data to identify new targets. So in my field, in this field, scientists have integrated all these data across studies to firstly to create consensus sub-alert. It's like Google map or human body. So you can query a gene expression, you query a cell types in this map. And then by integration of the data, we also increase the power of the downstream analysis, allow us to better characterize the patient-to-patient heterogeneity, also enhance our capability to identify rare cell types. These rare cell types could be associated with the drug resistance. So since we are entering the big data area for single-cell biology, so in other area, for example, we have AI scientists already developed large foundation models for big data. For example, ChatGPT, which is a large foundation model trained with massive language data, massive text, and then later adapted for different tasks such as translation or conversation. So inspired by the success of ChatGPT, computational biology have also developed AI foundation models to answer to tap on the large biological data to answer biological questions. So what is foundation model? It is very different from the canonical AI models. It is not a task-specific model. It is meant to replace the task-specific models, and it is pre-trained on the large unlabeled data, which is called large-scale pre-training. And after that, we fine-tune the models with small labeled data that could be in-house data for diverse downstream tasks. These downstream tasks could be like patient stratification, drug response, or prediction, or like biomarker discovery. And this model will be trained with self-supervised learning or transfer learning at these two stages. And many examples of these foundation models we can observe in the media, so such as a Solar, GPT-4, DALL-E, and the BERT. So what is single cell foundation model? So we are inspired by this ChatGPT, and then scientists are developing single cell foundation models. And these models adopt the self-supervised pre-training techniques and the transformer architectures, which is to leverage the success of the paradigm in the language and the vision models. And why foundation model? Because in my opinion, single cell big data is a gold mine. Gold mine is waiting for us to mine it, but we don't know how to mine it effectively. And data is huge. We have the single cell data of nearly 100 million cells, which constructs the, I would say that constitutes the universe of human cells, of all the human cells. And then we have many different downstream applications that can tap on these big data and tap on these large foundation models. So, but we are not the first ones. So there are several existing language foundation models developed for single cell biology, such as GeneFormer, SGPT, UCP, SC Foundation, and NicheFormer. All these methods has been published only this year or last year in very high impact factor journals, indicating that this is very trendy right now. And then, but if we go deep into all these methods, right, and then we find that there are significant gaps in these methods. And firstly, single-cell RNA sequencing data is fundamentally different from language. Gene expression is not sequential. It's not like the words "I love Singapore" that is a sequential order in the words. We don't have such sequential order in the gene expression. Secondly, expression values are continuous, so it's not like the words which is discrete. And last challenge is the data is very noisy and sparse. So we need to tailor or design the foundation models specifically to address these characteristics of single cell data. And at the largest gap, in my opinion, is that there are many foundation models out there, but they are not truly useful yet. So they haven't like really fine-tuned it for like a very useful downstream applications, which is very important for today. I mean, like, for us to discuss what is the application we can make use of single cell foundation model. So here with a team in A*, we try to build this large foundation model for drug discovery. And we first pre-train these models with publicly available big data, and then fine-tune these models with small labels data for drug discovery. And this small label data comes from either the in-house single-cell data from patients upon treatment. So we have the metadata indicating whether this patient is a responder or non-responder. In addition to these patient data, we also have in vitro drug screening data to find. With those data, we fine-tune the models to predict the drug response or treatment outcomes. And more importantly, we identify a cell type specific markers that are associated with drug resistance, because in the past, we only have the bulk level of biomarkers for drug resistance. Now we are going into the cell type specific biomarkers. And lastly, we like to predict or simulate the perturbation of the genetic perturbations response in the cells. And with that simulation or prediction, we hope we can convert a responder to a non-responder to a responder. So we can save a lot of time in those wet lab experiment. So this model will be trained on the DISCO database, which is, I think, is the largest and manually curated single cell data. And it contains both healthy and the disease tissues. So it's like it constitutes a universe of human cells across healthy and the disease and different tissue types. And then with these models, we focus on Asian biology, Asian medicine. So we pick two use cases. One is a liver cancer, another one is a colorectal cancer, which are both prevalent in Singapore, in Asia. So we fine-tune these models with the Singaporean data from these two diseases first as a the first two are use case. And then this model, of course, is tailored for single cell biology, the single cell data to adjust the non-sequential nature of the data. And lastly, and then I will talk about the RNA model, RNA language models, which was proposed by Miller. So, and then with help of Yuguang from IHPC, we extend these models to multimodality, which have three modalities here. Okay, so with the first modality, we use the tokenized methods to generate a text embedding from the metadata, drug information, and the gene identifier, which is the gene symbol or gene name. In the second modality, we use the variational autoencoder to convert or encode the gene expression into an embedding. And in third modality, we use a graph autoencoder to encode the prior knowledge of the gene interaction graph into a gene interaction embedding. So with all these three modality embeddings, we feed this embedding into a transformer, which is the large foundation models with pre-training for using a generative pre-training. And after that, in the fine-tuning, we use our in-house data generated by our collaborators and other researchers in Singapore to train these models to for the downstream tasks. The first very boring task could be like cell type classifications, but the more exciting would be the drug response prediction and key biomarker identification, and even like genetic perturbation response prediction. And most importantly, this model is explainable. It outputs this attention score, which allows us to identify the novel gene-to-gene interactions or gene-to-gene associations or regulations that are specifically enriched in one phenotype but not the other, such as in responder but not in responders. And then with this attention score, we can better identify the genetic not genetic, gene signatures that are associated with the responder, not responder. And then, so this model is, in general, is meant for cell discovery, and then we focus on Asian biology and we use patient data from Singapore, including the liver cancer treated with immunotherapeutics, as well as colon cancer patients treated with chemo. And then we use the single cell data to identify a cell type specific biomarkers for the responders, and also try to predict whether this patient is going to respond to a certain to the IO or the chemo. And on top of the patient data, we also have so I'm done. This is my last slide. So we'd like to deliver the foundation model pre-trained for drug response prediction, and that can also adapt for other tasks. And then we aim to identify new biomarkers for drug response. And then lastly, I'd like to highlight this. With this foundation model, we'd like to create a virtual cell in silico. In this virtual cell, we simulate the perturbations. So we don't need to do the experiment. We simulate in the computers like for certain perturbation. It could be a drug or CRISPR, it could be aging, and see how the gene will be changed upon this perturbation. Thank you.
You've given a very nice overview of foundation models and your own work. But I want you to, in 30 seconds, please, ask the question that I said, which is not about your own work, but where do you see this being placed in context in the field? How important do you think foundation models are going to be, not just in this space, but in other spaces?
Yes.
And second, is this the best way to identify targets for drug discovery, or are there other alternative AI-based models?
Yes, so foundation model can be applied to anything. So like for language, for biology, for finance, I think it can be applied to anything. The fundamental thing is the big data, a well-curated data. This is the first thing. So we have a lot of genomics data from the precision medicine, actually 50,000. And we also have the IDA data. We have a lot of data, not just single cell RNA sequencing. As long as we have a massive corpus of well-curated data, we can build foundation models. So foundation model means nothing. It means like just a machine can observe the knowledge from the massive data. And then, other alternative approach. Of course, there are alternative approaches. And then
I know obviously you will be a supporter of your own approach, but please set in context of the audience. What's the other approaches and their relative merits and
Yeah, so yeah, for now I don't. Yeah, I'm not AI. Also, I'm not a pure way AI. Yeah, so I tend to come out from the like more like what's the unmet need first? What the unmet need first? Yeah, yeah. Thank you.
And without further ado, ask
Okay, thank you very much. So my topic today, so first of all, thank you for inviting me to speak today. And my topic today will be on the topic of AI plus RNA. So because I'm in the Department of Pharmacy and Biomedical Informatics, I want to add a slight drug twist into my topic today. So me and Shyam has worked together very closely, and he's given a very good broad overview of basically everyone who's doing RNA in Singapore. What I'm going to do, and we didn't coordinate this, but it's almost like we did. What I'm going to do is to take two of your topics and drill down very deeply, and then hopefully we can come out on top and talk about what's next in the field. So this is a highly simplified overview of the drug development process, right? We start with target identification, and then we score some leads, and then some promising lead will be taken into preclinical studies, and eventually we'll have some clinical trials. So today, the two topics that I'm going to drill very deeply into is target identification and lead screening. So for the first one, in the past 10 years, I believe there has been a paradigm shift in the way that we discover drugs. So in 2010, if you go to a pharmaceutical company and you propose a project to them, it's most likely to be hypothesis driven, right? You would likely have done significant work in understanding the biological pathways. You would have likely have very clear idea of the knockout and knockdown studies, and you have done this in animals, right? But nowadays, if you propose a similar project to a pharmaceutical company, you would likely have come from a perspective of a data-driven science, right? And this is because of three thrusts that has really became the mainstream in the past 10 years. And the first one is human genetics. We have data sets like UK Biobank, and then now the SG10K and SG100K. And secondly, there is an explosion of single cell and spatial omics data, right? As Jianmiao has alluded before, there has been 100 million single cells deposited in the public domain, and there's much more, hidden in individual labs that's not being shared right now. And thirdly, there's machine learning, especially pre-trained models, to analyze all these data to extract meaningful information from them, right? So from the data-driven perspective, I'm going to drill down to a very specific topic, which is ancestry specificity. And I think the reason I choose this area to talk about today is because I think Singapore, if we want to position ourselves to be the leaders of the world, we have to position ourselves in an area that we have an existing advantage, which I think is ancestry specificity is one of those things. So this is a collaboration that we have done in the past 2 years with Shyam and Ken Hong. So this is the Asian Immune Diversity Atlas. So we have performed single-cell RNA-seq about 1 million PBMCs in 500 donors of diverse Asian descent. And then we have classified the cell types into major cell types. And here I'm just showing some of the ones that most people are familiar with. Dendritic cells, natural killer cells, T cells, and B cells. And what we have done is that we want to understand the genetic underpinnings of how splicing is regulated, and how that's different across different populations, right? So on the left-hand side, I'm showing the number of discoveries across different cell types. So as you can see, there's quite a bit of cell type specificity because the number of discoveries of splicing QTLs varies dramatically across different cell types, right? And also because we have 500 individuals this is the largest, to our knowledge, this is the largest single cell consortium for Asian descent. And this is a very large data set and empowered us to not only map cis-QTLs, but also a more challenging topic, which is trans-QTLs. And here you can really see the cell type specificity. So for example, the tallest bars only appears in a single cell type. And then the ones that are shared across 2 or 3 cell types are actually minor bars, okay? So I'm going to tell you about 2 stories that we are able to do because we had Asian biology. If we had, for example, European biology, then we wouldn't be able to discover these signals, okay? So the first one is on basic science. So what we have discovered is that there are 2 regulatory variants independent of each other in naive and memory CD4 T cells that regulates this protein called HNRMPLL or HNRFLL, okay? So this protein, so this is why it's independent, because we have discovered 2 independent signals. And this protein regulates the splicing of CD45. So I think some of you in the audience is familiar with this protein, CD45. Its isoform-specific expression is very important for T cell development, right? And as you can see that this regulatory relationship is highly cell type specific. It occurs in T cells, but not other cell types. And then we went one step further to do an orthogonal validation, which is, previously we have asked from a genetic standpoint using Mendelian randomization, but in parallel, we could also do this in a single cell type fashion, right? Which is not possible before. We can stratify the cells into whether they have expressed HNRFLL. And then for those cells that have expressed, we observe that CD45 splicing is high. So PDPRC is synonymous with CD45. And then for those who not expressed HNRFLL, then CD45 splicing will be low, right? And then we could even go one step further to say that because we have some prior knowledge about the role of CD45 isoforms in T cell development, are we observing genetic influence on CD4 cell type proportions? And after doing Mendelian randomization, we observe that that's indeed the case, right? We observe a very strong signal between the genetic variants that's influencing isoform splicing and T cell proportions. So the second part I'm going to tell you, so this really gets into the Asian-specific part, is the disease biology, right? So we observe that there's significant colocalization between the splicing QTLs that we have discovered versus autoimmune diseases, right? So here I'm picking one as an example, which is Graves' disease. So here you see that Graves' disease GWAS colocalize with different cell types, SQTLs, in a very cell type specific fashion. So it's high in CD4-14 monocytes, but then the signal disappears in naive B cells. And we got very lucky here because the variant that's influencing the splicing happens to sit on a splice junction. And this splice variant is specific to East Asians. So as you can see here, its allele frequency is high in Japanese, Chinese, and Korean, and it goes down in Southeast Asians, here Malays, and then it sort of disappears in Indians. So you observe this beautiful gradient between East Asians, Southeast Asians, and South Asians, right? And this is what we would have expected based on the FST values. And then you can also validate this using 1,000 genomes. It only appears in East Asians and disappears in other population types, right? So I want to drive this point home by saying that this is only possible because we're in Singapore, right? If we have done this in a European population, if we have done this in UK Biobank, we would not have observed this signal because this variant simply does not exist in UK Biobank, right? And then, of course, we validated this using a mini gene construct, and we observed significant difference between the reference allele and the alternative allele, right? Okay, so this is the first point. This is the point about Asian specificity. And the second point, as Shyam alluded before, we can design RNAs instead of screening them, right? So it used to be the case that we start with targets and then we have a library of small molecules that we just screen, and then we have perhaps 100 lead, and then we follow up on each of them, right? But that's not the case anymore nowadays, I would like to argue. The case nowadays is that we can design these molecules, not only small molecules, but mRNAs with very high precision. And perhaps we can just come up with 3 of them and validate them in a very in-depth way. So this is what we call lead design, right? So this is a collaboration that we did with industry. This is an algorithm called linear design. So I'm going to give you some background on mRNA vaccines, right? Or in general, mRNA therapeutics. So used to be the case that we target the proteins, but nowadays, because we know we can program mRNAs much easier than proteins, we can use these as the drugs themselves, right? And you can liken them as, for those of you who are computer science, we can liken this to code on your computer, right? So mRNAs will be the C++ source code, and your ribosomes will be the compiler, and then it compiles in your body from mRNA to its executables, which is the proteins, right? So what are the pros of mRNA vaccines? I think personally holds enormous potential because it has transient expression, unlike CRISPR, for example, which is kind of dangerous of the off-target effect. It's a transient expression, and it's less dangerous. You can use this with less concerns in humans, right? And also, it's very easy to produce once your targeted protein is identified. But there are some drawbacks of mRNAs. The most prominent of which is that mRNAs are unstable, right? It's single-stranded. There's a bunch of endonucleases and exonucleases. For those of you who do experiment, the first thing you go to your bench is to spray all over your bench RNase inhibitors, right? And you do this every half an hour because there's RNAs all over in the air and everywhere, right? And it's harder to transfect. So this is the mRNA that we used from the spike protein. And then, as you can see, there's cut sites in lots of places in the spike protein. And what we really want to do is to transform this highly unstable structure to this very stable structure, right? So you can see the reduction of the cut site from this structure to that structure. So I'm going to so the x-axis is the minimum free energy, and the y-axis is the codon optimization index. So I'm going to tell you how traditional drug companies are doing it. I shouldn't say traditional. CureVac and Beyond. 2 minutes, okay. CureVac and BioNTech and Moderna are brand new companies, but they're more traditional in compared to our algorithms. So what they are able to do is that they can optimize the codons, which is the easy task. But our algorithm will be able to optimize this along this area, which is a much harder problem, right? This area is entirely unexplored, and the feasible region has a possibility of 10 to the 632 candidates. In comparison, there's only 10 to the 80 atoms in the universe, so it takes a long time for you to optimize this. And after we optimize this, we can see that we have higher higher mRNA expression, much longer half-life for the mRNA. We almost double the protein expression. We increased the anti-spike antibody by 100-fold, and then we increased the neutralizing antibody by 10-fold. So this is a significant improvement compared to the current state of the art, right? And the way we did it is by a joint optimization of the structure and the codon optimization index. So you can express, I'm going to skip this quickly. So you can optimize the codon optimization index. This is easy. The hard part is you minimize the free energy. And then the key of the algorithm is that we use lattice parsing to make this from an exponential problem search to a linear search. This is why we call this linear design, right? So used to take years to generate these mRNA molecules, but we can generate these mRNA molecules in minutes. Okay, so I'm going to skip all these as well. So what's next? I think because we talked about two things in depth, right? On the discovery side, I think it's really, really important to have ancestry specificity. And this is, like if we're in Europe or somewhere, I don't think this is a big area to focus on. But because we're in Asia, especially we're in Singapore, where I think Singapore is the most diverse ethnically country in Asia, I think this positions us in a unique area where no other country can surpass that in a natural way, right? And the second one is that we want to sample diverse molecular phenotypes. Most studies nowadays focuses on mRNA expression. This is because it's very established. There's commercial methods to do it. But we also want to do, for example, splicing, chromatin accessibility, and also multi-omics, right? To make sure that we're sampling broadly and looking not just under the light of commercial availability, but also the darker areas, right? And this brings to the third point, which is joint analysis of multi-omics data sets. So that's the first point from the first study. And I think on the lead design side, I think there are two thrusts that should be followed up on. The first one is end-to-end optimization of full-length mRNAs. This is a problem that's very difficult and has not been addressed as of today. And the second one is we want to design circular RNAs and self-amplifying RNAs in the future, right? And this is an area that we are currently looking at, but we're very happy to discuss with any of you if you're interested in this topic. Okay, and thank you very much. And these are our wonderful collaborators who have supported us along the way. Thank you.
Thank you very much. Before we move to the general discussion, I'm going to challenge you with the second of the two questions I asked Jianmiao. You've outlined your vision based on the work that you and others have been doing. What are the alternate possibilities for mRNA drug design? Why aren't, for example, Alnylam and Moderna adopting?
So I think there are two ways to go about mRNA. So we have taken the approach of deep learning. We have done it in a very traditional natural language processing way, which has worked well for us. But then now we're at a bottleneck. We realize that there are some limitations. You see designing the coding regions, but then in the non-coding regions, you see that the rules that we're discovering using traditional natural language processing ways is very limited. And then you have to really do some unsupervised feature extraction. So I think for the non-coding regions, we really have to bring all the data and stuff of the deep learning area. But then we had a paper on this before, but I didn't talk about today, is that for some tasks, we actually realized that not deep learning the traditional rule-based ways is better than deep learning. The coding region being one of them is a highly constrained optimization, right? Because we know that we only have slight flexibility. We know that we have to make macros and only have slight wiggle room. So in those cases, I think rule-based is better than deep learning. So how do we merge the rule-based ways with the deep learning ways in a productive fashion? We don't know the answer to the same path.
That's fair. Yeah, thank you very much. Ask Jianmiao, Boxiang, and Shyam. You're welcome to drag up chairs for the panel discussions. What I'd like to do is just summarize a few of the points that we've heard in these really exciting talks, and then open up for questions from the audience. I see that there are a number of people, before I go further, I see that there are a number of people online who are saying, I can't hear this, I can't see this. Look, this workshop was designed to be an in-person participatory workshop. So if you have problems, it's not too late. Please come join us. So I'm going to summarize what we've heard. A poor summary, I'm sure, because we've covered a lot of ground. So the question that we posed really in this session was, in the realm of RNA and RNA sequencing, where can AI really and what are potential endpoints? So 3 questions that I pose to the speakers. I'm going to summarize what we've heard. But what is the progress that has led to the current state of the art? I think the recurring theme has been the availability of very large data sets which are spanning many different types of RNA data. They range from single cell data, bulk data, crossing populations, cross genetics, cross. Jonathan and others have also been working on RNA modifications, which is an emerging area which none of the speakers alluded to, but I'm. So first, we have a lot more, and we have a lot of that pertinent domain for Singapore. Second issue was, where can we find? What are the major future challenges? I think again, recurring theme, at least one, there is potential to apply AI. However, we need to be cautious, as Shyam in the type of model that we use. For example, neural networks may not be optimal in all settings. And I challenged both Jianmiao and Boxiang to say, you guys are advocating a particular approach, foundational models, or the type of linear design for RNA. What else is out there? What are the alternatives? And my sense is we haven't got a consensus yet, so this deserves discussion. Third, what can we do in Singapore? A recurring theme was contributing to drug discovery, identification of targets, which Jianmiao and Chow spoke about, and the optimization of RNA-based therapeutics, which Boxiang particularly spoke about. In addition, Shyam raised 2 other interesting examples where we might get together in Singapore to do something world-leading, which is in the area of early disease interaction, predicting outcomes, predicting prognosis, predicting who is likely to develop disease. So that's my summary. So please open now for questions from the audience. Please remember that the aim of this question session is to arrive at recommendations that Young and Kedar are white. So please speak up. Yes.
And very nice talk for both of you and also Chow. You know, you have quite much on the, using the RNA-seq, right? Whether single cell or bulk cell. And they said that very good at identify the novel drug target because maybe it's uncandidate. So if you give us a few examples where you find you can really things down, which is a real, an important drug target, which so far.
Okay. So far, we don't have such example of success yet. I just believe that the data is like a blue ocean. You need drafting into the water to the ocean and then trying to, with the AI or models to identify new drug targets. So for now, I would say, to my knowledge, we haven't been successful. I couldn't pick up, think of any example yet for now. But yeah, I'd like to add one comment about the foundation model, deep learning, blah, blah, blah. I think the major caveat is that these 2 disciplines, biology and AI, there's lack of communication, and we need more close collaborations to design a biologically informed AI model so that the model is explainable, interpretable in terms of biology. So I really would like to bring these 2 disciplines together, hold hands with both sides to design the best biologically meaningful AI model. I think this is the major caveat in the current AI, why AI is still not being useful for biology yet.
Jianmiao, thank you. I could agree more with that point indeed. I challenged Yang at the very outset. How does one set AI into context? Because it's a tool, and there are many different contexts. And I agree with you wholeheartedly that we need to get biology and AI holding hands. So this is an important point. AI has promise, but it hasn't yet delivered anything, at least from the experts and the speakers, it hasn't yet delivered
targets?
I think we have something to add to that question. I think both of us, yeah.
Oh, yeah,
Can go,
that's-
go first.
Okay.
Yeah.
Thanks for the question. So RNA sequencing is valuable, but spatial omics is also valuable, and a lot of it is imaging based. It's not sequencing based, but you're still profiling RNAs in the tissue. Okay. And we really believe that single-cell RNA-seq, powerful as it is not powerful enough to discover all the interesting biomarkers in those tissues. You need to see how the cells are arranged, and that's a spatial RNA profiling question. Single-cell transcriptomics, traditionally, you're dissociating the cells, you're losing the tissue structure. You no longer know which other cell this cell is near, because you've broken up the tissue into its constituent cells, right? So that's one point. To really be powerful, we need multimodal AI that connects or unifies or merges single-cell RNA profiling, spatial RNA profiling, spatial protein profiling, maybe patient metadata, and tissue imaging, about which we have decades of experience, right? So I think that's where the breakthroughs will come in. And getting to Ashok's point, because of the Singhel Digital Pathology Archive that has been accumulated over 10 years, digital pathology means just tissue imaging of tissues from patients. They have tissue images from patients for like 50 diseases, 150,000 patients, 1 million images with matched clinical data, medical records, right? That is a unique resource. Very few places in the world have that. And so we are very well poised to exploit that and add some high throughput molecular data on that. So that's coming to the competitive advantage. But the other question you asked was, have we found anything interesting, some potential drug targets, right? So indeed, we believe we have. So the pathway to finding drug targets is you do some molecular profiling. As Boxiang mentioned, we're going away from target candidate gene-based methods where you just guess, okay, I think this gene or this pathway is important, to just profile a lot of molecules and then see statistically which genes stand out. And then you call those biomarkers. Okay, so they have genes that mark some biological feature or clinical feature of your patient. And then some of those biomarkers are diagnostic biomarkers, meaning they tell you whether the patient has the disease or not, or they tell you what flavor of the disease the patient has, or they're predictive or prognostic biomarkers, meaning they tell you whether the patient will respond to the drug, or they tell you whether how the patient's disease will progress, right? So that's just a biomarker discovery. But once you discovered biomarkers and understood its biology somewhat, right? Some subset of those biomarkers will be good drug targets. So as I mentioned, we have already found very promising diagnostic biomarkers that are ready to move into clinical trials for chronic myeloid leukemia. And we believe we're in the early stages of finding some targetable biomarkers for colon cancer, right? Those cells that are budding off. And then I did not have time to show, but we found some cell-cell signaling interactions. 70% of approved drugs, not just cancer, all drugs, 70% of approved drugs target the cell-cell signaling molecules, the molecules that enable cell-cell communication. And spatial omics is fantastic for identifying these cell-cell communication interactions. And indeed, we found some novel cell-cell communication interactions that we believe may be driving some of the properties of these cells that are cancer cells that are proliferating. So those could potentially be targetable. It's not like we have something IP that we can immediately go to Merck and say, okay, pay us a lot of money for it. But I think we're, on a good track. We found a lot of interesting biomarkers just from our relatively small data sets. So we're very excited about how far we can go from the much larger data sets we're generating going forward.
Yeah, first of all, I agree with Shyam 100%. And I want to turn your question on its head. So your question is, how have we discovered any drug targets using AI? But I think the real question is to ask is, are we discovering too many drug targets using AI? So, right? So we have from 2010, from a discovery standpoint, now to a data-driven point. So for those of you who have run a differential expression, right? With disease state versus healthy state, you get thousands of genes. And each of one of these genes is a potential drug targets, right? So the question now is not to discover more targets, but how to hone these targets into actionable ones, right? And to that point, I agree with Sean completely. We need to do multi-omics, right? So if we just do single-cell RNA-seq, we'll discover thousands of potential drug targets. But if we combine RNA-seq with genetics, which is multi-omics, we hone down those targets to tens. And then if we further combine with spatial omics, then we can really get down to the biology, right? So one of the slides showed that we have the TCHP gene, which is a target, potential target. I don't want to oversell it, a potential target that's filtered through thousands of genes, and we really find this very strong cell type specific signal and also agent specific. And eventually we validated it using experiments, right? And I think that's a potential target that sometime in the future, maybe we can propose to pharma companies. Yes?
Well, that sounds very interesting. That discussion, we should move on. Thank you for the question, Jonathan.
One point mentioned third time also what unique aspect Singapore has. And having been here for more than 10 years, every time I go to conferences, one of the most striking things for me is in terms of technology, Singapore is very fast. If there's a new technology, you go to there or anywhere. Very likely Singapore has seen it either before or at the same time as someone else. Single cell sequencing very early, long read sequencing that I work on very early in spatial transcriptomics. We understand that Singapore is really very huge on. So I think that's a unique aspect. And so in a way, that's all captured under the RNA profiling, RNA sequencing. All of the examples that we heard about rely on this RNA profiling technology. And I think having this kind of early access to technologies allows us to develop communication methods like I'm doing or social media that really are essential was preparing the data in a way that all these methods using these gene expression matrices or anything else in terms of different gene expressions. So there's this huge amount of tools that are absolutely essential that can benefit also from AI. And there's very good examples of constructing genomes, very applications, many different applications for plants, gene economics, or anything else. They don't necessarily require a specific application. And so I think the point I want to make, which would be in your opinion as well, is that to maybe YouTube make for the space of RNA genomics and AI, one doesn't necessarily have to have an application. If you develop a method that underlying all of the applications that you're using, that can have a huge impact. Yeah, but it's not necessarily linked to an application. So I think it's good to think about this kind of space of AI and RNA that drives the field forward, that leverages on technology, but it doesn't necessarily have to have a very specific disease application. The RNA genomics itself.
Absolutely. Yeah. Thank you, Jonathan. Those are thoughtful points. And before I hand over to the speakers, I'm going to ask you and the speakers another question. Singapore is an early adopter of any new technology. Does Singapore persist with new technologies until things have been ironed out, or do we move from place to place without realizing the full benefits of new technology? That's a question I think for you and for the speakers.
Yeah, Jonathan, I agree and disagree with you. So Singapore, Singaporean, or everybody, us here, are capable of doing many things, but we are not quite sure what is the most significant or impactful things we should do for the best benefit of Singapore and Singaporean. So I don't tend to do these things because I'm capable of doing it. I need to ask myself, why am I doing it? Am I addressing an unmet clinical needs or medicine need in Singapore? I need to cater my research to serve a bigger society to see what actually Singaporean needs. And then I would love to involve like clinicians or like hardcore biologists into this discussion, like what they are really eager to know, what the challenge they're facing, what I would like to start from the unmet needs first. Yeah.
And I think in the third session, we will be addressing just this. May I ask you to comment on Jonathan's early adopter of technology? Are we also early leavers, premature leavers?
Yeah, jumping too fast from one technology to the next.
You think so? Are you agreeing or disagreeing?
I I am that kind of person, so I try to stay very yeah. Yeah. I think it's good to tap on the latest technology to stay cutting edge, even leading edge. I want Singapore researchers to be leading edge, not just follower, to be leading edge, to lead something, to invent the next edge. So I'm not a big criticism of the jumping from technology to technology. I embrace the leading the edge. Yep.
Maybe I can address that. So I've been here 16 years, and I do see some of what you're saying, Ashok. Maybe you've caught on much quicker than I caught on, despite having come here very recently. But there is a hype cycle, and sometimes through nobody's fault, we tend to follow the hype cycle, and actually a little bit late, meaning when the wave is crashing, then we go all in, and it's kind of should have gone in earlier. But then we see the wave crashing. As soon as we move in, the waves crash, we get discouraged, and then we run to the next wave. But by then that has crashed too, you know. I really feel that what we need to do as a community is build resources that other people can build on top of. And stick with it. Meaning if whatever wave I'm chasing now, it's my duty to create something that, when the next wave comes, this stuff I'm doing will still be useful. And that's why, for example, so I've stayed in single cell for 12 years. Because I see that it's gone from, oh, I'll get a Nature paper because I have a single cell data set from 2 samples, to it's a workhorse. But actually, for me, that's when it becomes exciting for actual translation, because now the technology is scalable, is cheaper, is reliable. We know how to analyze the data, and we can do powerful things now that we couldn't imagine doing or had no ability to do 10 years ago, even 5 years ago, right? So I really feel that we have to. So I won't get a grant for saying single cell now in Singapore, but I'm still putting it in my grant proposal, and we have to stick to it. But just combining it with the newer technologies, and hence the move to spatial and working with I²R on EHR, combining with EHR and stuff like that.
So in other words, evolving your approach along with the tech.
Evolving rather than abandoning.
Yes. Can I add some? Yes. I just want to add one more point. I think there needs to be pillars or centers of excellence that continues on the trajectory of one area of technology, right? So, for example, for Jonathan, it takes you perhaps a few years to learn the long read sequencing, and it takes us a few years to learn single cells, right? And I think it's a waste if we learn these things, because it's a steep learning curve. We learn these things and then abandon these after newer technology comes along. I think it's important for some labs to stay in a specific area and then really becomes the world leader in that area for Singapore, and then to support other labs who are coming into those areas, right? And I think that once we have some of these centers of excellence in that trajectory, then it's harder to abandon, because it's an economic of scale. Other people are coming in for you for help, and then you get more funding, and then you can continue on developing what you're really, really good at already, right? I think that's, and we're seeing this actually in Singapore coming along, and I think that's a very, very good outlook.
Thank you. Very interesting perspectives on an interesting question. Perhaps we should. Yeah, so we need to take this on board, and perhaps, Yang, this is an issue that we need to consider, particularly from the perspective of biomedical data generation. Your AI is going to depend on methods of data generation, examples we've seen in the RNA space today. So the data generation methods may change, but again, it may be necessary for us to build source assisting with some of these methods in order to apply AI well. This may be a sort of point to take home from the initiative perspective. Yeah. Next question. Sorry, who was first?
Boxiang first.
Okay, just a very simple question. Around 20 or more years ago, there was this sort of hype of biology, and people are thinking that we want be able to one day be able to model a cell in a computer and then of course actually do all kinds of things. And fast forward to today, obviously there are factors like complex. Okay. So now instead the idea is that it is AI instead. Now, but it seems to me like going back, why did systems biology fail? Which I think personally I feel that one of the things that perhaps it goes back to the because when you want to build models within the cell, you need a lot of not just right now. I mean, just cell RNA, but what about the proteins? So can't all of it. Metabolites, there's so much to build. So we can't really build a cell for it. So with that, given the current limitations within the measurements, how much would that affect AI tools that we're building, and therefore the output that we get from this AI would AI face a similar kind of limitations as what systems biology did 20 years ago?
Interesting question. So the idea is that there are limitations in the types of data that we can acquire in cellular systems in terms of dimensionality. Let me introduce, before I hand the question over, another dimension which we have not spoken about, particularly RNA expression. Cells can change in minutes or hours their RNA expression. What are we sampling when we do single-cell RNA sequencing? When we do different types of RNA sequencing, are we really getting the full picture, which I think is the so over to Jianmiao, Shyam, and Boxiang.
Okay, thanks, Koshon, for the question. Yeah, I used to work on system biology and work with those ordinary differential equations one by one. So I would say, I won't say it's a dying topic. So maybe in the future, we may have like more data, like maybe other modalities or maybe a new ways to reactivate or invent this area. So my comment is, I think the biology is very complicated system. With very complicated system with one data modality or with one methods. We won't, even for a human being, we can't comprehensive this complexity. That's why sometimes I feel that I'm so tired of looking at this complexity, so that I just say, okay, maybe AI can help me. So this is the challenge I'm facing. For example, even like for just gene expression, the single cell data, when I try to identify biomarkers from these massive data, I do encounter like many factors. For example, batch effect, confounding factors such as age, your ethnicity, your work style. So all this is too comprehensive for me to design to comprehensive this complexity. So yeah, that's when facing this complexity and this massive data, I feel that I'm kind of like yeah. I need to seek help from the AI for the
mathematical summation.
Yeah.
Individual data points is always there. But thank you for that perspective, Jianmiao.
It's a great question. I think you're feeling the crash of that, systems biology or synthetic biology wave long ago. But let me tell you, it has not crashed. It's been resurrected. Have you heard of digital twin? You must have heard of digital twins. Now everybody's talking about cellular digital modeling the cell. Chan Zuckerberg has this large grant program now, or research program, to do a completely model a cell and its perturbation response and so on. So the hype lives on. Okay. My personal opinion, and it's not just my opinion, I'm happy to see this. Some experts in the field also mentioned that. Same thing you're saying, same thing Ashok's saying. We're looking at RNA. There are so many other molecular layers in the cell, and those molecular layers aggregate into organelles, right? So the system is underdetermined. So I come from a physics background. My undergrad was in electrical engineering, so I did system dynamics, system theory, and systems modeling and all as an electrical engineer, feedback system, control systems, and so on. As a physicist, I modeled a complex system, which is a particle accelerator for my PhD. Those were still all relatively tractable systems. When I see the cell And I see people claiming that they can mathematically model the cell from first principles. I just feel like this is so far from reality. Because what you can observe is just one small slice of what's happening in the cell. There is no way from that you can build a de novo mathematical, dynamical model of a cell. Okay, so let's put that aside and as just hype. If you put digital twin in your grant proposal now, you'll probably get some money. Okay. But what's real, let's focus on what's real because we want to build something that we can be proud of 5 years later. I mean, still keep going 5 years later rather than jumping to the next topic because, you know, we got a grant for something we could never really achieve, right? What's real is you can still predict something about the cell, not from a first principles dynamical systems model, but just from some kind of curve fitting and extrapolation, which is what AI can give you if you have the right data. Okay, so for example, so Jay has this, Jay Shin, my colleague Jay has this program called Atomic, where the idea is to very high throughput throw a large number of perturbations at cultured cells and have a very high dimensional RNA readout. And then plonk that high dimensional RNA readout into a lower dimensional, let's say 30, 40 dimensional space. Okay. That's not such a huge space. And then with a lot of perturbations in that system and single cell readouts, you can really kind of explore the space of cell states that are created by all of these drugs and pre-drug molecules that you're testing, right? And then if you have a new drug that looks somewhat like those drugs, you can guess that the response will look like somewhat like that response, and the cell state that you're pushing the cell to looks something like the cell state of those similar drugs, right? So that's just in some sense a curve fitting or smoothing or something, but Jay is not trying to come up with a mathematical de novo systems model of that cell. So this kind of predicting cell behavior using AI is actually achievable if you have the right kind of data, if you have enough training data, right? And I think that's the kind of thing that could be practically useful.
If I may, we are talking about dimension in a rather loose sense. And I just wanted to make sure that, because much of the dimensionality that we're talking about is actually between interrelated things, such as, for example, DNA, RNA, there are clear known relationships between them. And in a way, sort of mentally envisioning the problem, you could say a lot of those properties of the cell project onto the same XY plane. But there are other dimensions, such as, for example, time, such as, for example, external environment, which includes all of these, which don't project into the same XY plane. So we have to be careful about how vision dimensionality, and it's just a point that I wanted to but before I hand over to the speaker.
If I can quickly respond to that point. You're exactly right. So when I said you project it into a 30 or 40 dimensional space, there are another 30 or 40 dimensions we're not seeing.
Exactly.
So it's quite coarse, but it's better than where we were before.
Indeed. And you're sort of doing a maximum intensity onto almost flat planes in order.
Yeah, it's a projection. Yeah, absolutely.
I just want to add one, or actually before my point, it's almost funny because Shyam was a physicist, I was trained as a physicist, and I know Professor Zhang is a physicist. So when we made the switch from physics to biology, when we do physics, we think that all we do is beautiful. And then when we switch to biology, what we do is nothing but, right? It's ugly. And it took me some, and I think I can speak for Shyam as well. It took us for a long time to get from beautiful to ugliness. And then now we see the beauty in the ugliness, right? So we didn't get to talk about that point in my slide, but what we're seeing is that different omics aspects would tell you completely different stories. So what I showed us is splicing QTLs, which if you had looked at expression, you wouldn't have discovered the same signal, right? And I think we can propagate this into other omics. So if we only look at expression and splicing, you would miss chromatin accessibility, you would miss protein methylation and protein post-translational modifications. So the point now is to collect as many dimensions as possible, including time, for example, and then to build just to curve fit using these multidimensional data. And I think I think the AI tools are already mature to do that.
So we have time for one last question before we start to bring this discussion to a close. The question and the answer is a bit brief because we only have 5.
Thanks. I think it's a fantastic discussion. So we've seen the genetics, the DNA, the RNA, proteins, right? What we've noticed is that there's not much common over that. So now that we know that cohorts being collected, if it's precise or cohorts, is that a benefit in looking at those cohorts but asking these questions rather than, because we know I'm an immunologist, but don't collect the right sample, no matter how many samples you have, right? So should you focus at least 1 hour on current cohorts that are being collected or going to be collected, and then in a specific manner ask the questions with a low-hanging fruit, because they have correct samples, you have to do the omics. So is that something?
So it's a good question, and it's a very practical question, which Yang and Keta might need to take account of. Should we focus on in AI on data sets where there is already history in Singapore collections, such as SG100K and some of the efforts that our speakers put? Brief answers, please.
Yeah, I agree with you. We should, make use of existing data sets first before we expand to new data set, integrating the new data set, because any one additional layer of data modality, it will increase the challenge and difficulty in the data analysis for like bioinformatics, for statistical analysis, even for the almighty AI is also another challenge. Although we have these multimodality foundation models, these foundation models are going to multimodality, but I think that we need to take one step at a time. Yeah.
So short answer is absolutely yes. Like 2 more sentences. You want what are the requirements for a successful program here, right? You need a large cohort, like a good number of samples from a good number of people. You need a lot of metadata from those people. So of course, as you know, data analysts, we're not going to go out and create our own cohort. We have to leverage existing cohorts. That money has been spent. A competitive advantage is exploiting that data and augmenting that data with some more data generation. Precisely what we're doing in IDA, we're generating our data from the SG100K cohort. AI for drug discovery, we're generating our spatial omics data from the Single Digital Pathology Archive cohort. So all that metadata is already there. The IRBs have been approved, the samples been collected, and we're just leveraging all of that. Otherwise, it's very hard to compete with the rest of the world.
Precisely, I agree with the 2 speakers. In order for us to compete, we have to not only leverage, but also combine. I think, like, for example, what we're doing in IDA, it's not possible for a single group to have that much money to do it. And same thing for SG100K. I think all of Singapore have to come together to make these pillars of excellence.
Great. So I think we have an answer. I'm going to make a few remarks as I bring this to a close. We just have 2 minutes left before coffee. So I think the first session has been fantastic. At least it's been mind-opening to me. So thanks again to Jianmiao, Shyam, and Ashok for really excellent talks and really excellent discussion. And my thanks also to the audience for listening and asking very penetrating questions. So we have spoken in this session, in this theme, about RNA data collection and its potential for analysis and outcomes. We've heard about foundational models and certain other paradigms which could be used in the AI space. One question I'd like to address briefly to Yang is, Yang, from your perspective as an AI specialist, here is an area. What are we missing in terms of AI science in order to analyze this?
Point out a very good point. It's that I want to give my remarks at the end of the closing remarks, but data is very important. And I will emphasize to also invite discussion about the RNA structure prediction. It's a very good example of kind of importance of the data set. So this is a part of the thing I have seen. This is somehow miss the view that's not. That's how we, curate data, how we really, with the limited data, how we develop better or smart algorithm to still train on model.
So the data curation is happening at the level of all of these efforts, SG100K, the new RNA initiative, they're all curating data. So I think that element is being taken care of.
Yes, and also the RNA really recognize.
Of course. But the question is the last point. Where are we missing the AI science? Do we have all of the know-how, all of the components to develop new and better smart algorithms to take advantage of that? Yes. You think we do in Singapore, or do you think that there are holes?
This is not an easy problem.
No, that's why I'm asking you.
The community try very hard. And also, I consider myself like an AI for science scientist. So we are also trying very hard to develop new model, very specific question. I will talk about this in more detail.
That's good. So we will wait for that. So I'm just going to bring this session to a close and just make a few. I think this is an example in a wider biomedical context about how data accumulation around one dimension, in this case, we focus on RNA, and through the application of AI, lead to and help us to obtain particular outcomes, drug discovery, early intervention for the two themes predicted here. And I think we have a number of points of feedback from the community and from the speakers behind it. Thank you all very much. Coffee break now. Thanks.
Excellent.
So sweet. Very sad about it.
It's now yes.
Hello, everyone. I'm Polly Chen from Cancer Science Institute, NUS. I'm very happy to be the chair for the second session. So the first speaker in my session, sorry, my program not updated. So is Dr.
Mu, yeah.
Dr. Yuguang Mu from Nanyang Technology University, and he will be talking about share the overview of AI-based RNA small molecule modeling. So I hope that you can talk a bit about your own research, and then spend a bit of time to talk about your vision about the AI-based research in Singapore, and maybe how you can contribute to the community. Thank you so much. Yeah.
Thanks for the kind introduction, and thanks for Ashok, and also for Yang, give me a chance to talk here. Lai Heng is my student, and in fact, we all in mind, right? Most drug is usually working on the protein side, which receptor or GPCR or kinase. In fact, the protein is not only the target. In fact, RNA is also a very important target. In fact, in 1940s, the drug streptomycin, which is working a very popular, antibiotics, which working on the bacterial ribosome. In fact, RNA side, and also quite a few of antibiotics we found similar way. But there's one very interesting drug which is FDA approved 2020. It's called the risdiplam, which is working on the RNA splicing. It's the small molecule which working on the intra and extra the interface, which can help to solve some missing splicing issue. So it's very and so this such FDA approval or the real function of this small molecule, right? In fact, inspire the field, which we cannot only focus on protein. In fact, RNA is more have more abundance and also more a type.
not only the extron, also the non-coding RNA side. It's a lot of, I think 10 times or even 100 times more than the protein types. So that's why the RNA targeting molecule can be a very good tools to target so-called the undruggable disease or undruggable protein. In fact, the binder of the RNA can be different types. For example, there's a HRV, the TAR RNA, which is well structured, and some molecule can bind there and have some anti-viral infection effects, and also some so-called undruggable protein, which we can target on their mRNA side, which also have some structure, for example, the loop or the mismatching. Then there's also some very clear effects, and also the RNA associated pathway, which also can design small molecule, for example, ribbon switch, can especially and affects or modulate the function, and then we can help to do some very interesting the pharmaceutical effect. So now, what is the challenging? I think we all know that RNA is quite flexible and also very hydrophilic molecule. The backbone have a very negative charge. And usually small molecule, right? We like hydrophobic pocket. When the pocket is too hydrophilic, it's difficult to design very good drug. And also the structure compared to protein in the PDB data bank, they are very, very less RNA structure. I think Yang will talk about later how they develop AI tools to predict the RNA structure. And also there is the limited nuclear base, only 4 types compared to protein 20 amino acids, right? So the diversity or the specificity may be a little bit a issue. So of course, then if you have a lead, right, or heat, how to do heat to lead operation, also a little different from the protein side. So this I think we need to clearly consider. So here I just copy from the very good review, is that you see there's a few stage which we can somehow think about to use RNA targeting small molecule drug discovery. The first stage definitely is the target identification. I think the previous session, which 3 speakers, I think very to talk about using RNA-seq or other multimodal omics data. We can very good a way to identify which RNA sequence or which gene have can be a good target. Okay, then we do the hit identification, which try to, of course, my talk mainly focus on the small molecule side. Then we can use some AI-based screening to do the hit identification, which is my mainly research area. And then we do the following the hit to lead. Maybe it could be borrow many years of experience from medicinal chemistry knowledge to do that. So I think the first one to do, how can we apply AI to, right, in the first stage? How we can identify very important and unique, also maybe personal medicine related target. This, I think we can use many tools for them, even ChatGPT to analyze all the literatures and also combine with RNA-seq, genomic data, metabolic data, and then together we try to find a pipeline to analyze, to pick out which is the most significant of the drug target. And then we need to find out the target RNA disease association. Why you think this gene is more important than others, and why it could be function as a drug target. This, I think, the AI tools definitely can be applied here. Okay, with the big data, with biological literature, because many data, right? Many biological funding is separate or distributed in broad literatures. So we need, some basic language model to do that. Another way, if we find the target, right? If, for example, those target usually is undruggable for protein, then we can go to RNA level to see whether the RNA level can be a target. So we need to do the structure prediction. And this, I think we do not talk too much. I think this will be, I think, definitely talked by Professor Yang. And also the next step, right? After we get the structure, can we quickly and efficiently find the hit molecule? And this is what we, what my research. For example, recently we published RMSD XNA, which including RNA-DNA, and the platform for RNA targeting small molecule water screening. And we use, in fact, not a very deep learning model, but the XGBoost, the model to, of course, the feature we use a little bit interesting and unique is the physical inspired feature, which is the distance based. We base on the structure. So here I show you, for example, the general protocol. For example, in this case, right, we take the molecular one, which is already well dissolved 3D structure of non-coding RNA. And then we use the nearly 2.3 million small library. And then we choose we choose top 20 ligand. Then we do the MD simulation. And you see, I'm not sure can show very clearly. So some drug, right, in simulation binding very stable, but some quickly in during simulation, I think around 6 months they flew away. So this molecule we discard. Then we buy, I think 10 compound, and then we do the cell-based, validation. And this, I think we try 4 cell line, and 2 compound. It turned out very efficient. For example, micromolar, and even the one compound 10, even is I think is 100 nanomolar in the tank 1 cell line. So it indicated, right, such targeting, right, really we can find some hit. And also we also do the preliminary data, right? We also do some fluorescence check whether this small molecule really bind to the on target, really bind to this RNA. It show out. It's very interesting is that the 2 compound, right, the effects on the RNA structure is different. One compound is destabilize this RNA structure. Another compound stabilize. It's found out that the destabilize is more potent. Yeah, I think this is quite interesting. So another thing, right? We also found that we, in order to get a very good hit rate, we need to have a focused library, especially for small molecule. And then this, of course, some big chemical company, for example, Enamat, they already have such, focused RNA binding library. I think that is quite useful. In fact, so that help us to, quickly to screening, and then to come up a possible hit and reduce the false positive, because any prediction AI based or whatever based, we want to reduce the false positive. Yeah. So then the next stage, right, hit to lead optimization, which definitely we have to combine medicinal chemistry knowledge and also the binding, the detailed binding pattern. For example, how the small molecule interaction with the RNA side. I think this crystallographer may not be very helpful here because RNA quite flexible and the crystal structure, right, it's not easy to get. But I think NMR, the NMR technique maybe is best suitable here. And so I think that the challenging is that, for RNA structure prediction is that we have a limited data. And also for RNA ligand interaction side, we also not many. So that's how. But now we also, I think, of course, here I only show one story, right? But if we consider all whole Singapore or whole grand project, right? We can definitely combine all the effort. For example, from the upstream, the target validation, which you mentioned that you already find a good target. Then we can do the following. If protein, then we use the protein side. If RNA side, we can also use the RNA target score function. Then we do the hit finding, then hit validation, then hit to lead. So I think this whole platform, I think, is feasible. At least from my point of view, it's really we can target some very maybe Asian specific disease or other, not easy handle. What we tried for cancer cell, right? Pancreas cancer cell so far is still so-called to be unmet the disease. We can somehow to focus on that direction, which other technique, right, is so difficulty and all RNA targeting small molecule may have a chance. So I think that's all for my talk. Thank you for your attention.
Thank you so much for about his own research and his opinion about what is the most obstacle in terms of us RNA targeting small molecule research. Singapore. Shall we do the question at the end? Okay, so then I will move on to the next speaker, Professor Zhang. He is actually a newly recruited senior PI at CSI and also professor in School of Computing, as well as professor in Department of Biochemistry and US Met. So I think he's very well recognized for his groundbreaking discovery, kind of using AI tools and bioinformatic algorithm to predict protein. And now he's heavily working on RNA structure prediction. So Prof. Zhang, please. Thank you.
Yeah, thank you. Hello. Yeah, thank you, Polly, for the introduction. Also, Ashok wanted me to talk about the AI and RNA structure prediction. So basically, I want to quickly go over, because I only have 15 minutes. So go over about, what happened in the field of RNA structure prediction and where are we? So the definition of RNA structure prediction is very simple. So basically, you have a one-dimensional RNA sequence. So how we can use computer program to predict this coordinates of the three-dimensional structure. So as Panis mentioned, my lab or the field, actually structural bioinformatics, has been focused on the protein structure prediction for many years. RNA structure prediction is pretty new. The reason is that, RNA, the biology of RNA is just, recently recognized. So this is a chart which is kind of motivation for us why we move to this field, because the drug in this entire genome. So traditionally the drug target is protein, but protein only count on 1.5% of the human genome. So because RNA have very important biological function, and so if we are able to model the RNA and design, actually many of them are drug target of RNA. So the magnitude of this future, if we can make it a RNA druggable, probably increase more than 10 times. So because of that, And recent years, the field of the RNA structure became hot. So in general, there's two types of the computational method for RNA structure prediction. One is traditionally using the statistics or homologous modeling, comparative modeling. Somehow it's based on some physical law. And this is new. This is AI-based structure prediction. So this is I taken from one of my students which developed. This is one of the first AI-based method for RNA structure prediction. We are using query sequence and then use end-to-end prediction transformer and also protein language RNA language model to finally get this RNA structure. So when we talk about the RNA structure, we have to talk about a very important Olympic game in the field. So I know many of you guys know this, but in case you don't know this, I will briefly introduce this. So this is originally from the field for protein structure prediction. So now extended to RNA structure prediction. So basically every 2 years, an organizer collects some of the sequences, nobody knows the structure. Then they send the sequence to the structure prediction and also send the sequence to the structure biologist, asking to solve the structure. Then this is a blind prediction. So 3 months later, the organizer will compare your predicted model with the experimental structure and then to check what is the current state of the art of structural modeling. So this is the newest, this actually is the first time Cas include RNA. So Cas15, this is 2 years ago, and there's only 12 target. So this target will have 3 categories. Some are easy. Easy means you have template in the PDB. Some are difficult, you don't have a template. And there's some of this protein or RNA are designed by computational program. So it's called non-nature human designed protein, RNA. So those are the best model, which I will show you here. And this is a superposition of the predicted model with the experimental structure. So you can see they are doing pretty well. And those are easy target. And these 4 are human designed target. So this RMSD means the higher the worse, the lower the better. So there's some difficult target, very big, and without template, there are, RMSD is pretty high. That means we're unable to follow them. So if we look at the AI method, we found that AI can do well on this natural RNA. But for the non-natural RNA, they are generally is bad, very bad. Actually, this is understandable because designed RNA, they don't have homologous in the library. So, and your machine learning, you can learn only the pattern from natural existing protein, RNA. So I will talk about this later. So this is kind of the ranking of different methods. So here you can see those are with the deep learning. Those are like actual top method are traditional statistical or physical-based approach. And on average, they are outperform the AI-based approach. So that is quite surprising because if you look at the protein and the statistical method is this. Here, TM score higher the better. And this is AI combined ITaser. There's a dramatic improvement of the AI on protein. So especially if you look at the CASP, this is every 2 years. So this time, this is a traditional method. And the TM score higher the better. You can say majority of the protein we cannot fold. They're below the TM score below 0.5. So when So the success rate is 10%. But when we introduce deep learning to the field, you can see we have much more successful. So especially the last case, Thematic of the or majority of the protein, we have very high score. So if close to 0.9 means they're almost like an experimental structure. So the success rate is from 10% to 91%, just because of the introduction of AI. So the question is that, so here is one of the very exciting examples. So everybody knows the AlphaFold, and they can, so those are just, I show the structure. So why AI is so powerful for protein? One of the major reasons is that if you look at this, Chen is a traditional MD simulation, and they only have like 20 parameters in the 7 energy term. iTaser is a knowledge-based approach. There is 100 parameter in 26 energy term. So basically, Chen cannot fold any protein. And iTaser can fold some protein, but in the medium resolution. But AlphaFold, they have 10 billion parameters. So that's actually, to me, is a major reason, because biology is complicated. You cannot really use a statistical method which can describe using a few parameters. But machine learning, they use a huge number of the parameter. They can really learn into the the specific energy, structure pattern. So, but let's come to the AI. Why AI cannot work as well on RNA as on proteins? So I think there are several reasons. One of the major reasons is that, the PDB, if you compare the number of the structures in the PDB and a protein, you have this, I already split into different chains. So there's more than 600,000 of structures. And RNA only have about 4,000. Those are redundant. If you remove the redundancy, you have about 1,000 RNA structure. And a protein, you have 97,000. So that is, we know that AI is based on learning. So they learn the model or train the model from the natural experimental structure. If you don't have that structure for them to input, to train, how come you can make AI work? So that is partially which Ashok just mentioned, why the data set. Data is really important. And that is one of the important reasons of this AI not work as well in the RNA. So you can see this is how RNA has structure has been solved. They are increased pretty fast, but the number of this type of the RNAs is still very limited. The second reason is the sequence databases. If you look at the protein, because protein nowadays we have meta-genome sequence. So currently the sequence database have more than 35 billion of the sequences for protein. But RNA, we only have, 18 million sequences. Number of the sequences is very important for derive this, co-evolution, because co-evolution is a major input feature for machine learning. So if you don't have a large number of sequences, you are not able to build a co-evolution model. So that is also the, disadvantage of RNA. The third one, probably actually just now, some people mentioned about this. Our protein, they have like a higher heterogeneity. So they are, because they have 20 amino acid, and probably from long time evolution, they have probably a better, well organized, the landscape. So they are more stable. But the RNA, they are, they only have their energy landscape is less, so it's more dynamic. So that's a part, is also part of the reason for the, if this is a well-folded structure, so computer is probably easier to fold. So given this, disadvantage about the databases, also the structure itself, so we really need a new AI strategy to address that kind of issue. We cannot use really the traditional or common AI method. So that is already demonstrated in this case of competition. So we have actually, I have some idea. So we're thinking, because Ashok also asked me what you guys really develop new methods to address this issue. So I just, I'm open to welcome, you guys give us some idea, but this is what just I'm thinking. So one way is that, we cannot just either use AI or physics, but why not we can combine AI and physics? So this is one of the way we are trying to extend the ITaser to RNA, and we are using deep learning and plus the physical force field and do the simulation. Looks like they are working pretty well. And the second one is that we're trying to use a diffusion model to, to suppose this is a native RNA. We try to diffuse that, and then we generate many, almost infinity of this, the conformation, decoy conformation. In this way, we are able to use a network to learn, the pattern from the computational decoy, so that can partially address the limitation of the data set. So those are just some of the idea. So now I come to the concluding remark, is that so AI works well for some of the RNA, so they are doing better than traditional method, but they are feel difficult to predict the new fold, including synthetic RNA. So apparently the AI-based protein miracle has not yet been seen in RNA, unfortunately, but I'm very confident this will happen. So the challenge is that structure and signal database is too limited. And also the RNA is more dynamic than protein. So the challenge, we have still, opportunity is that we can, have like a uniform the algorithm to combine advantage of both AI and physics. We also, here, this is a challenge. We also really need a new idea, new AI technology to overcome this data limitation. So somehow we think the word find in protein, that we find a language model is useful to deal with this sometimes the limited data. And diffusion model is actually is a novel method, has not yet been used in the proteins for structure prediction. So diffusion model, some of the lab is using for the protein design. So this is a very promising approach we are trying to work on. So hopefully we can communicate with AI scientists, we can get a more newer idea. So that's all. Thank you.
Thank you, Prof. Zhang, for the very nice talk. So let's move on to our the last speaker in this session too, Dr. Roland Huber. He's a PI at GIS and I think he has a very strong expertise in study structure elements in human and the viral RNA. So Huber, please. Yeah.
Thank you. Thanks. Yeah, very good afternoon, late morning. Can you put it on full screen, please? I very much like the talks, and there will be some overlap in terms of the topics that I will address. Professor Ashok asked me to talk about three things, basically, like what are we doing at the moment, what are the challenges that we are facing, and what I think we should be doing. First, I want to start by motivating why do we care about RNA? We care about RNA because intervening on a protein level is relatively constrained. We have a limited number of proteins that are expressed. We have a limited number of those that are druggable. And then we have interfaces, we have complex regulatory chains that are very difficult to address with small molecules. So if we can modulate RNAs, if we can directly address gene regulation on the RNA level, we would be very successful. We can also introduce new genes with RNA. So this is a vector for gene therapy, it's a vector for vaccination, and that's very interesting. And then also we have success stories like small molecule drugs that are able to bind and modulate RNA. What are we doing at the moment? I have to speak a little bit about what we're doing at A*STAR. There is obviously an initiative that's been shared by Shyam before. This is Mile is very crucial in leading this effort where we're looking at language modeling, transformer models that have been very successful in natural language processing. And the idea is that to basically we replace things like multiple sequence alignment, where we learn the evolutionary context of one sequence, with things like language models that ideally contain the evolutionary context of all sequences. And then we can use these embeddings to learn complex features about RNA and do things like predict secondary structures and predict tertiary structures and predict other ancillary properties that these RNAs have. We are also looking at small molecule targets. So this is a project that we recently started. This has been spearheaded by Patrick Dunn as part of the precision medicine and is now led by Van Yue from GIS. So we are looking at targeting RNAs that are relevant in gastric cancer. We are also looking at manufacturability. This is part of the Welcome Leap program, is trying to establish foundries for RNA. And the idea here is that we investigate what types of sequences are manufacturable, what motifs are problematic for manufacturing, how can we avoid this, what motifs are important for high expression levels, and so on. And then there's also a component where we're looking at RNA ligand complexes, where we're looking at all the complexes that are available in the PDB. There's different binding modes. We are obviously more interested in specific binding modes. We also have unspecific binding modes where we're looking at things like intercalation or groove binding that are not really sequence specific very much. But we want to have pockets, or we want to have whatever that means in an RNA context is still up for debate, or still something that's being investigated, like which sequence and structural motifs of an RNA are targetable, and how do the types of ligands look that would target something like that. Is also an important consideration, as Professor Zhang just mentioned. What are the key challenges that we face in this area? If we look at, if you want to use RNA for therapy, whether as a target or as a drug, then there are certain challenges that we need to address. So if we want to use RNA as a target, particularly as a target of small molecules, then tertiary structure prediction is a crucial element that we need to address. And I think we have just seen that there is a lot of progress going on in this area. The other type is what types of ligands bind these tertiary structures. The second challenge is if we want to use RNA as a drug. So the rollout of these mRNA vaccines in the COVID context has shown that this is can be a very effective strategy, and that this is in general quite a safe intervention. There are certain issues that also have been raised before. One of the issues is that these RNAs, if you introduce them, have a relatively low persistence, which for a vaccine is okay. But if you want to use that in the context of gene therapy, you need like systems that are much more persistent or self-amplifying and so on. And then the third challenge is manufacturing these aspects, and there are issues like how do you scale that up? What are features that are detrimental, that you don't want to include, that are immunogenic, that are cause strand breaks, that cause impurities? All of these things at the same time while optimizing other properties. So if you look at RNA as a target, then yeah, one of the key challenges is definitely that there's not a lot of structural information available. There's a few thousand structures available in the PDB. Very few of those are RNA ligand complexes. There's a few hundred of those. And non-redundant structures, so this is fairly limited. For protein structures, these AI approaches have been proven relatively successful. So one of the things that I think is crucial to advance in this area with AI-based is either combination. So we are using physics-based simulation as a data augmentation strategy, but also the combination of AI-based potentials and physics-based potentials to do structure assembly is a very interesting way forward. Yeah, what types of ligands is just underexplored, and there's not a lot of public data available. At least I know there are some initiatives in the private sector going on in that direction, with people building curated libraries of RNA targeting ligands. These methods, AI methods, have also been proven successful in ligand-based drug discovery, particularly graph neural networks for predicting all kinds of properties of small molecules, and it might be useful in that context. Yeah, and one of the other things that we did is look at virtual screening tools, docking algorithms and so on, and how they perform on the known RNA binders, where we have a structure and where we have multiple ligands that bind the same structure. And we find that there's some adjustment that is necessary, that the present tools not always perform optimally for RNA ligand interactions. And that's just because they have been fine-tuned for proteins, because that is the major bread and butter for the companies working on these tools. If we look at RNA as a drug, then there are basically two classes of that. One is using RNA as an expression platform, where we want to get the cell to produce some sort of a protein. And that might be for vaccine, that might be for purpose of gene therapy. So what is crucial here is having effective delivery, having a long lifetime, having efficient expression, and these are all properties that are both related to the types of sequences you put in and to the LNPs, how they interact as well. We are currently exploring, including different UTRs, for example, what effect that has on impression. Yeah, for gene therapy, you need something that is persistent, is self-amplifying. If we want to use RNA as an active compound, like as RNA interference, then there's a different set of considerations that also plays a role. So you have generally much smaller targets. There's few siRNA-based therapeutics that have really made an impact on the market. I think there are 3 at the moment that are FDA, or 4 that are FDA approved. And the major challenge for this has been delivery, and there has been some advances in that, but having something that is specific to cells would be very beneficial. And then also engineered RNA might be interesting in the sense that you have like environment-dependent riboswitches or ribozymes that can sense a cellular environment and then respond in a particular way. And that's something that we also working on with colleagues, where you have certain activity that only gets activated in the presence of other cellular RNA. Yeah, manufacturability, we've been working for the last couple of years within this SLEEP-R3 program with a variety of manufacturers to try to see what are the bottlenecks, what are features of these sequences that make a problem for large-scale production. There are challenges that are shared with other biological compounds in terms that the purification and the analytics is relatively complex compared to small molecules. And if you want to have successful drugs, you need to do a multi-factor optimization early on in this process, because changing everything later on requires you to go back and do safety assessment and get your processes recertified. So we need to have a way to predict early on, do we have problematic immunogenic, Do we have problematic production? Do we have problematic PKPD? Do we have problematic low expression features in the sequences that we intend to put in a patient? So it's a complex multi-factor optimization, and I think AI-based approaches would be very well suited to address that, but it's challenging to find the comprehensive data sets to do that all at the same time. And so one of the things that we did within this program is generate a number of such data sets and that we can work with. What are the things, pretty quickly, how we could move forward? One of the challenges that I think that is interesting is RNA dynamics. We know that RNA can occur in different states. So here, for example, you see a state that occurs in dengue viruses. You have a different topological organization depending on the context. These cannot coexist at the same time. If you interfere with one of these sites, you see a decrease in viral fitness. So obviously this is functional. But one of the things that we keep in mind, we often think about it, or this is the structure. But one of the things that becomes very clear if you look, for example, at experimental structure probing data, is that within living systems, RNAs exist, they change their structure, they change their topology depending on the cellular context where they're at. And I think understanding which regions are responsible for that and which regions are functional and are potentially interesting targets would be crucial. The other thing is we observe a variety of RNA regulatory networks where we have interactions between also we looked at this in a context of viral infection, but you can also look at it in a context of cell differentiation. You can look at it in a context of cancer. You have host-host RNA interactions or cell-cell RNA interactions. You have host-virus RNA interactions in this. And some of these interactions are pro-viral and some of these interactions are anti-viral. One of the things that we did is we collected a virus infection data over the years and we find some shared motifs. We find some shared host RNAs that tend to interact with viral RNA. And we are curious about that in terms of, is this a host defense mechanism? Is this a virus adapting to a host? So there are, we can observe these RNA-RNA interaction networks. There are a lot of cross-linking techniques that allow us to quantify this. And this data is available for a large number of cell types. Would be interesting to analyze and try to predict which types of RNA level regulation is relevant here. Another challenge that we observed also in the context of using RNA as a pharmaceutical in the SARS-CoV-2 pandemic is that currently the formulations are relatively cumbersome to handle. Degradation is a major problem. Thermostability is a major problem. You have these cold chain requirements where you have like to store your things at -80. It's very expensive. It kind of excludes certain distances that you can deliver that in less developed countries and so on. So one of the things, and also the RNA is relatively unstable in cells. So it's not unstable only on a shelf life, but it's also relatively unstable in cell and get degraded quickly. That can be a benefit, but can also be a problem. So one of the things that we're looking at is how do these degradation processes occur and which are the motifs that inhibit this degradation, both structural and sequence motifs. So we collect the structure probing data on a variety of, we have a library of millions of these RNA oligos, and then we see where does degradation stop, where does synthesis stop, and what are the features that are associated with that. For example, this is a well-known synthesis stop motif. It's called a type 2 terminator, but the same happens for degradation. And then the third thing, or the final thing that I want to show you is basically the structure inversion problem, where you have a certain structure that you want to design. You want to design, for example, an aptamer that binds a particular protein or that interacts with something else in the cell. And what you really want to do, you have a given structure, and you need a sequence that folds into that structure. I think there's very little information on something like this available at the moment. I think this is something that is potentially further in the future than many of the things that we discussed before, but I nevertheless want to raise that as an interesting challenge where we might have applications in terms of developing biosensors, developing specific aptamers for certain purposes. Yeah, and then I would just like to highlight there's a lot of people in A-stars, many of them are here today, who are working both experimentally and computationally on RNA, and I hope you get the chance to interact with some of them and ask us any questions. Thank you.
Okay. Thank you, Ruben. So can I invite the three speakers? Is it just standing here and do Q&A?
You can also bring chairs up.
Yeah,
I think we can stand for a few minutes. You want a chair?
you want to sit?
I'm fine to sit this way.
I want to sit.
Chandra and others who are joining online, this conference was actually, workshop was designed not as a virtual, but as a physical workshop. So I'm afraid we don't have set up systems for asking questions online. But as I said to the audience earlier, it's not too late to come and join in the very exciting discussions. We're only 15 minutes away.
So any Jianmiao?
Yeah, thanks for the nice talk. I learned a lot. So although this workshop is about AI for science, and then I would like to think the other way around. Whether there's something like a science for AI. So for example, in biology or in manufacturing science, we may encounter a new data type or new challenge and new questions that can inspire the invention of new AI. Maybe that all the existing AI models is insufficient to address this emerging challenge from the specific domains. So maybe you can inspire the hardcore AI experts to come up with the next generation of AI model. Maybe it's XAI or something. Thank you.
Yeah, this is exactly why we have this AI for Science initiative, because we want to bring together AI expert and the scientist. So that is also the question which I have, in my research, I really find that just I mentioned for RNA structure prediction, even for protein, we meet some very challenging, problems. We want to feedback that to the AI scientist. So unfortunately, I'm also affiliated with the computer science department, so I can directly communicate with people. But you raise a very good question, and also in the May 10th, there is a Science of AI workshop. So I hope you will be here and many other people. You can raise up your question. They are very much want to listen to what your question, because raise a good question is also very important, inspiring for the method development. So, yeah.
Okay, I completely agree. But for me, right, I can see mainly as application, not AI scientist, right? So we have very determined question, like Yang said, for example, protein RNA folding, which we have sequence and we target the structure. So the one-to-one mapping is quite clear, but the issue is how we get the new AI tools to solve. My area, for example, the RNA ligand, the binding, which also have a lot of data, a few data, then we predict. So we just try to use available AI tools to solve. So far, right, I feel is that when we have very clear target, then the data also available. That in that scenario, right, I think AI can help a lot when the data driven. But some area, for example, really talk about the digital twin, which want to simulate the whole cell or even how human body works. I think at the moment, I don't think it's successful because we don't know what the human how it works. there are lots of things we to know. Then how can rely AI to think about that? I think maybe a future direction, but I don't, at the moment, I do not believe that. Yeah, listen.
Yeah, I think that's a very important point in terms of models versus data. And I think a lot of the time, the thinking is that we can compensate for lack of good data by clever models. And I'm not sure how accurate that assumption is, because even if you have a very well instrumented model, if you feed it not enough or not good quality data, the patterns you will learn will not be accurate. And that's, I think, one of the key considerations, and I think it's also something that people should think about earlier on in the design of their research studies, is get the people who do computational biology, bioinformatics involved early on to think about what is the type of data you want to collect. How would you design an experiment so that you get the maximum amount of information out of that? I think that should be a very important consideration.
I have a somewhat deliberately provocative proposition in the realm of designing, predicting RNA structure and designing ligands. The proposition is as follows. I would suggest that virtually all coding and non-coding RNA species in mammalian cells do not exist in free forms. If you consider mRNA as an example, from the time of transcription, splicing, transcription, nuclear export, most of this is occurring in the context of protein complexes that are moving along the RNA. It's being spliced and transported as it's being transcribed. It moves to the cytosol and is immediately engaged by protein complexes that transport it for translation in ribosomes, where it then engages the ribosome machinery. Many non-coding RNAs, which exist primarily in the nucleus, exist as RNA-DNA or RNA-RNA complexes. So I would propose that actually there is probably no such thing, or very limited free RNA species in human cells. Now, this gives rise to a huge issue for a lot of the algorithms that you guys are developing, which Roland alluded to, which is the problem of adaptive change in structure determination. If this proposition is correct, a lot of RNA structure will not be determined by individual molecules, but will actually be represented in the energy landscape with hills and valleys and possibly even plains or plateaus represented by interactions between your target RNA, proteins, and other RNAs. How are you going to solve this issue? Because adaptive change, even in the protein-protein interaction field, has proven to be a really difficult problem to solve, particularly given the flexibility and degrees of freedom in the RNA folding landscape. Sorry for the long question, but I hope I've set it out clearly.
Can you repeat the question?
I
have a quick. I think I have a relatively quick answer to that. One of the things, I think all of the things you mentioned are correct and are a factor in the structure of RNA that we observe. And for this very reason, I think it is very important that we incorporate measurements into these models. We have a lot of techniques that allow us to probe structure of RNA, at least secondary structure of RNA, relatively high, in a high throughput manner. And one of the things that we are doing is trying to incorporate this information into both secondary structure and eventually tertiary structure prediction, because I think one of the key aspects is, you have protein interactions, you have different cellular compartments, you have different stages in the cell cycle where you have different host proteins expressed and so on. And all of that has an impact on structure. Also like different splice variants and things like that if you have a long range contact and topologies. So I think incorporating experimental data, people sometimes want to predict this experimental data as a proxy for structure, because it's been shown that if you incorporate it, it improves structure prediction. But I think that's backward. Because there is additional information about the context. The difficult thing that I see is that even if you look at single cell level, then you still have a blend of different structures that you're observing at the same time. And disaggregating individual molecules and understanding where they come from and what structure they have in their context is going to be a challenge.
Okay, I thank you for the question. I think what Ashok is want to ask how we can use the AI model to consider the environment and how the RNA and the RNA is not free. It's not a food by its own. They have to interact with protein or other RNA and environment. That's a great question. And before that, because I only answer your question based on AI because we're doing AI for science. So let me first introduce the protein-protein interaction, because you asked about that. For protein interaction, traditionally the way is very limited, only for protein-protein docking, physical-based docking. We can do very limited on, especially when you have binding-induced conformational change. So AI, especially the newer version of AlphaFold multimer, they make a great breakthrough on this field. So the current protein interaction complex structure prediction is much more accurate than docking. So what I want to say is AI play a very important role in protein-protein interaction. So you are right. We are just starting to consider protein RNA complex. This is what we're working on. And I believe the AI can do better than physics or than traditional way, because we already see from the preliminary data, we already see that especially if you train this like a language model for protein RNA interaction language model, that really work. I mean, it's more powerful than the physical based. So I think I'm very confident about this. I think this is what AI can make a difference.
Absolutely. Totally understood. May I ask a quick clarification? So one of the challenges, as I said, is, if you will, the degrees of freedom with a relatively flexible molecule like RNA. So how are you incorporating entropic parameters into RNA folding algorithms also to account for interactions that we've just discussed?
Yes. Because previously we were doing the single chain RNA, probably there are more dynamics. Entropy is really important. But when protein and RNA form a complex, they are more stable.
Correct.
So that is probably one of the good way for the AI modeling. So currently, AI actually, they don't care entropy. Entropy is important when you do the like physical based phosphate, you design your phosphate, you need that.
Exactly.
Yes.
So that was the reason for the question, because I suppose one of the I hope that AI will solve the problem, but one reason for my a dampening of my enthusiasm is, unlike proteins and protein-protein interactions, I feel encompassing the entropic parameters will be much more important for RNA and therefore much more difficult. But that was the point. Right?
Okay.
Yeah, I think your complete good point is that in the cell, everything interaction with each other. Especially, for example, for the MALAT1, the case which I studied, right? I look at all the literature, there are many one interaction partner, but I do not find some work to explore the normal function of that. But in the cancer cell, a lot of interaction. So what we are trying to design small molecule, which may be binding well and block the interaction with other partner. I think maybe this is one reason that it works. So we definitely, I think we use this, interaction network and tend to some interfere, then has an effect. I think that is maybe the answer. Yeah.
I think also something you mentioned before in terms of if you consider entropy you consider in terms of a conformational or phase space ensemble. And I think the approach of using diffusion models is very well suited to represent something like an ensemble, because you have you basically have some kind of a constraint, and then you have sampling from a space, and that might very well be able to represent all the states or the important states enough to get that. So I agree with you that you getting one structure, one answer, unless it's a very stable complex, probably not enough. But I think this can be addressed.
This is not a question, but comment and also to share the experiences that we have over the years. I think because RNA, other than RNA enzymes, most RNA do not need to have a structure for it to function, right? Because basically they are transferring the information to protein translation. Then in the context of drug discovery, where most often your targets are protein coding RNAs, right? So, and these are the ones that don't really need to have a function to have a structure to function. Then, so what we did is, we do a lot of coding RNA modulation as a drug target. So what we did is we use a physics approach where, because this coding RNA, when they are in the cytoplasm, while they're being transcribed, splicing occurs, 5' capping occurs, poly-A tail occurs, translocation to the cytoplasm occurs. All these are highly dynamic, right? And throughout this process, there are many RNA binding protein and even RNA and some fragment of DNA that bind to your target RNA. These are highly complex. So what we did is we used a physics approach, brute force approach, to kind of predict all kinds of structure that our desired RNA will fall. And although experimentally there's no way we can characterize the transient structure in the cell, right? So the only way we can read out is whether, if there's any molecule that we design to bind to our RNA, does it really work? Right? So it's kind of a surrogate readout. Then the biggest advance that we got that really get us to a very high accuracy of designing some oligo to bind to your target is when we solve the simulate the RNA structure, we actually even include structures that are thermodynamically not stable, and your Gibbs energy, is very high. So that means they are not supposed to exist in nature, right? But if I consider those then the accuracy of designing some molecule to target the RNA, the accuracy increased many folds. Yeah. So the biggest disadvantage of this approach is computationally it's not tractable if your RNA sequence is more than 2,000. Then you need to have hundreds of thousands of CPU nodes just to solve all these structures. Yeah.
I understand we're not recorded unless we speak into the mic. May I quickly ask Dave you a question? You say that there are situations in which you have unstructured RNAs which have worked as therapeutics, but do you know that they are unstructured in cells because they could engage the environment or other macromolecules?
So sorry, I don't get your question.
If you use or find relatively unstructured RNAs, which have efficacy in as therapeutics, could they be structured when they go into cells, although unstructured outside before you administer them?
Okay, so I clarified that the structure in a sense is because we are considering all possible structure of the target RNA, which is transient, I included those unstructured, right? So it doesn't mean that the vast steady state static structure is unstructured. Yeah, just to clarify.
Yeah, I think now not clear that how the small molecule, how interaction with the RNA, especially as you mentioned, the RNA structure is very transient. Some their theory that this molecule is a degrader, which to help degrade the RNA, make it even more unstable, then have some effect. But some small molecule stabilize. For example, some study on the RNA G quadruplex, which RNA, right? Some repeat then form a little bit stable G quadruplex structure, like a DNA. And those small molecule binder usually stabilize this G quadruplex. And then after stabilizing, their translation usually is halted. So in this way, so there the many different interaction pattern. I think we case by case. I think this.
Yeah, thanks to the three speakers for the fantastic talks. I was going back to one of the previous points mentioned by Ashok here about the entropic effect being taken account for the RNA or RBP binding complex. I agree. Also mentioned by Professor Zhang that the currently AI combined with physics-based is very important. I guess AI can give us some crude model, but when we can follow up with physics modeling to cover all the different aspects from the entropic perspectives. So here I have another question. Actually, if we think about protein-based, target-based drug discovery, it has so far gone through about 40 years. And indeed, the whole field has not seen a really breakthrough in terms of structure-based drug design for protein itself. Then in the case of RNA, I would imagine it's even more difficult, given that the structures for the non-RNA is quite little. So indeed, I was actually also wondering, before we promote AI for science, perhaps structural biology is indeed another important which we will never forget about. But again, talking about the approved drug for RNA, the risdiplam, for example, we know that there's one drug already targeting one RNA. So can we do something differently, not to be so ambitious like target-based for RNA drug discovery? Can we do something reverse? Like we know the structure for how risdiplam binds to its own RNA target. Can we start some prediction on fishing out those highly likely RNA structures that can be similar or close to the risdiplam's target? From there, we know we have a starting point. We modify the structural information for that ligand on the targets that may share some similarity to the risdiplam structure. So from there, we can start at least something slowly and gradually to have some rational component to be involved. That's only from drug discovery perspective. Thank you.
Okay, I think that example, right, 2020 is a FDA-approved drug. In fact, I think it's not rational design result. It's checked by some experimental chance or something like that. So that drug especially modulate the splicing. In fact, recently, right, after this discovery, there's some commercial or company, right, they just using the ligand base to, based on the scaffold of small molecule, then they try to build a library, a big library of similar molecule and see whether the same molecule have a similar, splicing modification property. There's one direction. But I think, right, in the future, in another direction is really, can we really rational target this splicing modification? I think the structure. I think that is a very good, because many disease relate to the splicing. Yeah. So I think this is a very good question. Yeah.
Yeah, I think it's a very interesting suggestion, and I think it goes into the sort of inverse problem in like give me all the sequences that look like this and bind this drug. Yeah, I think that's fundamentally a good approach, and but I think like from my understanding, structure-based drug discovery is used ubiquitously. Like it's not. You were very dismissive of it. I don't know. Like I have some friends who work in different pharma companies, and they all do structure-based drug discovery. I mean, with varying levels of success, but.
But if I may, Roland, on that point, I've also worked with a number of companies, particularly Aztecs, for example, where structure-guided drug discovery has really been pioneered. But I think, as was mentioned earlier, a driver in that is experimental structural biology, not structure prediction.
Yes, agreed.
So follow that, actually, I would just want to clarify. I remember probably Prof. Zhang or someone tell me that if the structure is predicted, like RNA or protein, if you further you to predict for drug, it's like it will never work, right? So at least one side has to be actual structure. Yeah, this is, I just clarify.
Yes.
You cannot use predict RNA structure for virtual docking or prediction for drug targets or for drug screenings, this kind of.
Yeah, I listened to your comments, but I don't really completely agree with that because traditionally, drug design rely on protein experimental structure. That's true because we don't have AI. AI become popular or especially powerful only in recent 5 years. So they need some time to field it. And especially for some of the target, you don't have experimental structure at all. And also AI now are not only predict structure, also predict the binding affinity and the structure, the interaction.
Totally.
So
yeah, I totally agree. And like everyone else, I hope that AI will solve these problems. But having worked for 20 years in small molecule ligand discovery using X-ray, cryo-EM, and fragment-based as well as other methodologies, I actually agree with the person who asked the question before, because I think there is a place. I'm sorry, we don't know each other.
Yeah.
But the fact is that you may be able to predict, but at some point, the experimental determination of structure, we've alluded to many of the issues, structural dynamics, the adaptive change that may be inherent in the RNA folding landscape, consideration of entropy. A lot of this in the end, one has to move from prediction towards experiment. So while not trying to dismiss in any way the importance and future of AI, I just think we have to be building capabilities in X-ray, cryo, and other new methods, even micro ED, to actually really be able to capitalize on AI. And I know from my limited knowledge of what big pharma and biotech is doing, they're all combining experimental with the predictions. It's only the really small biotech which don't have the capital or the capability to do the experimental along with the predictions who are investing in new algorithms. But I feel that at some stage, they too will have to get experimental data in for a lot of this. That's my view anyway. I may be wrong.
We need to really look a longer run because AI just a start. And you are right. Currently, AI models still need experimental validation and it's a prediction. But we just started like 5 years. What about 10 years later? What can you imagine? What the power of AI? Probably we don't need this experiment at all. That is highly likely. So this is the time and the opportunity which we should stand out and develop AI because otherwise you also when other people demonstrate that we are about other people. So
I agree we need to develop AI, but at the same we need to balance it.
I think one of the key aspects is that you demonstrate that your whatever technology you have is effective. When cryo-EM had 30 angstrom resolution, nobody was using it for small molecule discovery because it didn't provide any useful data. When you get down to 5 angstrom, it becomes more useful. If you get down to 3 angstrom, everybody's using it because you don't have to make a crystal anymore. And I think it's a similar thing here. Once you get to the level where you're confident that your predictions are accurate, and that you can rely on them, and that you can invest money based on that. And that's the ultimate sign of trust. If somebody says, okay, your model is good enough that I will buy these things and test them, and 80% work, or even if 10% work, it probably could. So it's really getting to the level where people develop this confidence. And the only way to get there is to do and to validate and to see and to demonstrate that you can achieve this level of confidence.
It's that point. So that is recorded. But it's that point that I'm really on about. I agree with the optimism. I agree that we need to invest in AI because of the promise, as Yang says. But I just feel at this stage, particularly in Singapore, which I think was the point that was raised, we should not neglect, at least for the immediate future, investment, for example, in structural biology to be able to get AI to the point when it can work without experiment. We're not there yet.
But I want to add one quick point. I have a solver. I test a solver. It's used by hundreds of thousands of people, and I have seen a very large portion of the people user are structural biologists. So computation, of course, our IT solver is not yet as accurate as now, but we have actually our new solver is even more accurate than AlphaFold. So my point is that using the structure global fold is one way to use that by structural biologists or biomedical study. There's many different ways to use the predictive model. So they can use that even to help them to solve the structure or to build a global fold of this architecture. So there's many different ways, even now. So we can certainly imagine this, I think AI and AI, even now, AI structured bioinformatics is complementary with the structured bioinformatics.
I think at the moment we should clear that how definitely we cannot not use AI, but we cannot see AI is 100%, can do everything. No, definitely. I think from my point of view, right, AI definitely can speed up or can make accuracy on the early discovery stage. But later on, for example, we can quickly find a good hit in diverse way, right? Then from hit to lead or even clinical, I don't think AI can help. It definitely rely on, but when they really the digital twin works, maybe we hope. Okay.
Okay, thank you so much during the limited time. So the dinner is ready outside. So lunch. So the next section will probably start between 1:20 to 1:30. So you have about 15 minutes for lunch. So please join me to thank this wonderful three speakers again.
All right, so we'll kick off the third and final panel ahead of the closing remarks. And so this area will be in AI. Oh, it's because it's LED. We'll be in AI applications in healthcare. So I'll be chairing. Jason is unfortunately under the weather with laryngitis. And then I'll also give the first talk. And hopefully I'll try to keep it really broad and then indulge me for the last 3 slides of some of our own work and how we're applying some of the problems here. So the idea, I think it's all very clear now throughout the day. AI is everywhere, particularly in cancer diagnostics and treatment. There's a number of different companies now, pretty much across the entire pipeline from fundamental cancer research, as we talked about in the earlier first two sessions. And now for my own talk, I'm really going to focus more on cancer diagnostics, where really we see most of where AI is truly impacting healthcare right now, right? And basically the two areas were brought up a little bit in some of the earlier sessions. One is now, Shyam actually really brought up a really great point about digital pathology really being an area that Singapore can contribute to. And then obviously, biomarker discovery, using genomics, and both areas, whether it's medical imaging or analysis of genomic or transcriptomic data, all of these can be used towards a number of different applications, from improving diagnostics, early disease detection, especially for like early cancer detection tests now that are starting to come online, like Lucentis, predicting patient health outcomes like Miraxis, and then precision guided treatment. Hopefully, companies like Orastone and others will come on board. So when we think about where AI in healthcare sits now, I think medical imaging is where it's most developed and most matured. So for instance, with Siemens, they have a wide range now of FDA-approved AI medical imaging software as a medical device platforms, all built around this AI RAD companion, which now basically is used to take existing medical imaging data, enhance it to allow clinicians to see more information, and then help them to make better decisions. Locally here in Singapore, I think this is where some of the technology that's being developed here in Singapore can really improve digital pathology by essentially taking images that we've seen now, and how do we extract as much information out of it, and information that maybe the pathologist or the radiologist cannot necessarily pull, right? So I took two recent examples of two startups here in Singapore. One is HistoIndex, which has really made a lot of gains in the area of liver biology, using their platform to basically quantify liver fibrosis, fatty liver, and now they're also moving into HCC, building using AI and ML-based stain-free digital pathology to extract features and then use those features to quantify them and create clinical predictions in terms of severity of disease. And then in last year in one of the EASL, they showed how it can be applied now into liver cancer, where they previously have had quite good success in liver fibrosis and fatty liver and NASH. Another company is Quirtive, which is quite successful for instance, they received their Class C IVD certification from the HSA for their QAI prostate diagnostics module. The idea here again is how can AI quantify medical images and pathology images to gain more information. So here, this is an AI-based prostate cancer tumor classification where it very quickly can identify and predict and outline for the radiologist or the pathologist potential prostate cancer tumors, even though they may be small or hard to visualize. So I think the idea here is really how do we use AI to basically be a better eye than our human eyes can do, and how does it quickly quantify and give us a numerical value for those, right? So the other area is this idea of how do we use AI in precision medicine, especially moving now towards companion diagnostics, right? So this is, I think, where we think about where most of AI in healthcare has also really been implemented, this idea of taking genomics. And now we talked about with some of the other groups, and hopefully this AI and RNA workshop has emphasized the need for also adding RNA to this, but other omics for developing biomarker-based precision medicines, the idea where we can take various different layers of these multi-omic data sets and then use AI to very quickly develop either panels like NGS targeted sequencing panels to either identify specific subtypes that may benefit from one treatment or another, or disease prognosis, or even, as we talked about in the earlier sessions, use this information then to also identify biomarker-based potential novel therapeutic targets. And obviously there's a number of companies, both local and international, that are applying this, right? And I think this is an area, again, this idea that it's very well established, right? So, there's a lot of NGS panels. Clinician can order the test now. The foundation panel is very well established, where it identifies a specific mutation, links that mutation to a specific drug treatment, and then the odds of that patient responding to that treatment goes up, right? However, one of the main limitations of this is that by focusing on a single modality, in this case, genomics, it limits the population in which this will be effective, right? So the recent review of the NCI match trial has shown that while the number of actionable mutations that are identified by these NGS panels has gone up, so now it's about 37, almost 40% have an actionable mutation. Unfortunately, the overall response rates of establishing a genomic biomarker to an actual treatment is relatively low. So overall response rate is only 10%. There are some winners in the group, but a lot of them, it's even if it's a biomarker and a drug that's attached to that specific gene, they're seeing overall response rates of like 0 for some of those drugs. So obviously, how can we improve that? And one of the ways is by implementing AI as well as other modalities like RNA to improve precision medicine. And obviously, I think the highlight of Singapore's contribution to this field is obviously miraculous with the development of GastroClear, where there was out of NUS and NUH was this idea of really having high quality quantitative measurements of serum microRNAs, and then using those data sets from those studies to then apply machine learning to then derive an effective microRNA gene panel. And then they very nicely showed that it outperforms clinical biomarkers. And as a result, GastroClear was then approved for IVD use by both in Singapore's HSA as well as in a number of other regions globally as well. And I think this really is kind of is a very good case study for how we can take very interesting technology locally and access to data sets that we talked about. We talked about how Singapore has a unique resource of data sets, how we can apply those very quickly to improve healthcare using AI and other modalities like RNA. The other area, and this is kind of where we get into some of the areas that I personally am interested in, is this idea that, and we talked about the function of time or the function of moving away from static models, is functional precision medicine. The idea that taking live cells, live models, and then and perturbing them, and if you perturb them in the right way, potentially you can then start getting information that whether you call it functional precision medicine, other people are calling it functionomics or phenomics. There's a lot of different terms for this, but it's really becoming an emerging field with some really great studies now coming up, and also ultimately, how do you combine this with the other data sets like the genomic and the transcriptomic data sets? So our own work in collaboration with Dean was this idea of using small data AI for more resource efficient analytics to really interrogate that phenotypic or functionomic space. And the key to it is really to get the right data, right? And the idea is if you have the right data in terms of very targeted phenotypic tests, then you can really quickly fill in your algorithm and identify, potentially optimal or effective drug treatment options. So in the area of how we apply this in healthcare, we've done a number of clinical studies. The most mature is in non-Hodgkin's lymphoma, where we were able to apply the platform prospectively to patient samples. And now we're showing that applying this platform in this way to guide treatment does offer clinical benefit. And we've had some really great exceptional responders. That's also been great to see patients actually benefit from this technology, right? And then, of course, as a result of this, NUS, this company has been spun out of NUS and is now has a MOH clinical license to run this test commercially, and is offered as a LDT to all clinicians, private and public, as of May 26. So this was actually the first MOH approved lab to offer ex vivo drug sensitivity, LDT, for oncology. Obviously, antibiotic drug sensitivity has been around for decades, but according to MOH, this was the first approval for this LDT here in Singapore. So to summarize, basically this idea that I think AI has been well established for digital pathology and precision medicine, biomarker development. But some of the current challenges really are how to use AI to make better use of the new modalities like RNA. There were some talks about metabolomics, Orstone, right, epigenomics. These are all starting to come on board. And then more importantly, beyond that, you know, phenotypic, and then Shyam talked about spatial, right? And then how do you ultimately integrate, you know, these? And I think this is also where it's going to be really difficult in the healthcare industry or in the healthcare field is you're going to get all these different new modalities and these new platforms. How do you really integrate them into clinical standard practice, right? And the biggest challenge here for these developing these molecular tumor boards that these are becoming increasingly complex, and your average clinician, how do you make them, what they want is they want to get this information, but they want it in a time efficient manner and as cheap as possible, right? And they want to have it in a clear way. So how do you do it in such a way that doesn't waste their time, allows them to continue to see their patients, and also doesn't cause decision paralysis, right? So what can Singapore do, right? So island-wide multi-institutional programs to develop and commercialize emerging tech. So the idea of working together across the different institutes. Examples in the public sector include, you know, things like LCG and CRP programs, and then also 65 labs, I think, is a great example of where we can start going to translate out of the public sector and into the commercial side. I think maybe Dean might talk about how, in order to kind of answer the biggest challenge here and integrating everything, how we really need clear coalitions for industry, clinical, regulatory, so that all parties involved know what a clear pathway to clinical adoption is. And again, this idea of how do we not waste clinicians' time, right, or cause decision paralysis, having access to electronic medical record sandboxes to really understand and give academic researchers and industry researchers a real understanding of how their AI platform and how their modality would fit into the clinical setting is probably really critical. Because if they don't understand that, if they don't have an idea for how they can be implemented into the clinical infrastructure, then it won't happen and it won't be adopted by healthcare. And it'll just be this cool thing that it was a Nature Medicine paper. Yeah. All right. So with that, I'll end and move on to the next speaker. Oh, sorry.
I know that the section was at the end, but I just want to clarify. I noticed you have not mentioned AI applications, image processing, radiology. Was that deliberate?
Oh, so that was, I guess, part of the beginning part of HistoIndex, Cordav, medical imaging. And I think that still falls under the idea of how do we integrate all of that? You know, how do we integrate the genomic or transcriptomic panels plus the medical imaging plus hopefully functionomics as well? But ultimately, there's going to be we're going to need, in order to really impact healthcare, a way to integrate all those data sets into something that clinicians don't have to spend 2 hours analyzing. Yeah. So next up is Professor Dean Ho, who is head of BME, as well as director of the Institute for Digital Medicine Wisdom, as well as N1, who will talk about digital medicine.
Thanks. Thanks for having us here today. It's good to see everybody. So I'm going to show very little data today. I think I'm going to try to summarize a lot of the questions and learnings that arise as we think about how AI is being increasingly discussed and hopefully integrated into everything from drug discovery and development all the way to healthcare. And I think there's a lot of misconceptions about when certain pieces of the workflow should be brought in. And a lot of it is followed chronologically. I think today I'm hopefully going to add some insights based upon feedback from the community on where I think some of these discussions should take place. I'm going to preface this with the note that what I'm going to talk about today will span from kind of the fundamental discovery and development side all the way towards implementation and policy. About 4 weeks ago, we had the immediate past FDA commissioner of the US here. And I spent some time with him talking about when people should engage regulators. And the mindset of our institute is that it should be early and often, regardless of the application. It can be a drug, can be software, et cetera. And and by the way, for those who don't know, the immediate past FDA commissioner is himself now an entrepreneur, right? And I think we very much saw eye to eye on that. About 6 months ago, I spoke to the current FDA commissioner, right? So now between the two of them, we're covering the last many years, right? Because the current FDA commissioner served under 2 presidents, right? So I think the feedback I get there is that people assume that they shouldn't engage regulators until it's time to get regulated. And the challenge there is a lot of people have to start over, all right, when that meeting happens. And so one might think that drug discovery and development, you don't speak to them until it's time to take a drug to market. But the answer is I think people should speak and engage stakeholders a lot earlier, all right? I'm going to fly through a lot of this. Just to set the stage, our institute runs a lot of trials. And these trials are all interventional, right? They're prospective, involving various ways, including different trial designs to validate different drugs. We've worked with pharma on this as well. And I think that we've learned a lot of what we do through these discussions very early on. Something that Ed alluded to about kind of frameworks to engage developers, drug hunters, medical device developers, et cetera, is what type of discussions do we need to have to ensure that the ideas we come up with make it to people and they make it to market. And I always give Yuan a shout out here, but and I'm going to do it again. So the first person that we brought into the Institute for Digital Medicine is Yuan Sapinel, who's on the right side there, top left, who comes from insurance, right? And we often get asked, why would we bring an insurance person who is not an academic into an academic setting? And the reason for this was because we wanted to make sure that the innovations we developed had a real chance to make it to people at scale. And in academia, we use that term a lot, right? Let's scale up, let's reach things at scale. But I think it's very hard in academia to really know what that means, right? And how to actually get something there. And so a lot of times we make assumptions of what that means. The reality is, in the end, nothing we develop, because we're talking about AI, we're talking about drugs, we're talking about devices, will ultimately be paid for by the patient, right? At the end of the day, someone else has to pay for it. And as we're talking about drugs today, there are increasing pressures on drug pricing and making sure that drugs are more accessible, to make sure that the trials run on these drugs are inclusive of communities. Okay, and the FDA is now and has been adding more requirements for the populations that are exposed to the drugs and recruited for these trials, right? So it's becoming increasingly important to think early about this. And people like Yuan are here to provide those insights and evolving insights as they emerge, and frankly, to kill projects before they start when we feel that, given the resources that we have, we need to make sure we understand what scale up means and that we have a means of building a roadmap to do that. We have a lot of implementation scientists, behavioral scientists. Matthias Eggermark, who's at the bottom there, he's a clinician, a practicing clinician from Roche, who has done Roche Pharma and Roche Diagnostics. And at one point, he was the only Roche global executive in Singapore. He actually attached with Wisdom for about a year, has just moved back to Sweden to become the GM for Roche there. But again, provided a lot of real world insight into what it takes to bridge an idea, right, with actual validation and what pharma looks for, et cetera. Right, so I'm going to fly through this. A lot of what we do is looking at small data, as Ed mentioned, but in the context of drug development and discovery, it's that we view each person as a dynamic subject, that the optimal dosing for a person changes over time, right? And using population approaches to derive dosing, even for trial design, not just personalizing medicine downstream, needs a rethink, right? And this was a fairly abstract concept 10 years ago when we proposed. But if you flash forward, this has really moved into things like the ASCO educational book, right, which is used to train clinical oncologists on a global scale. And it's a publication reserved for drugs and approaches that are practice changing in nature, right? And I found it quite ironic, right, that 10 years ago when oncologists told us to go away, 10 years later, now the workflow is in the ed book, right? And I mentioned this also because when I spoke at ASCO last year, I had a chance to field questions and engage with pharma to see how their mindset has changed in the context of clinical trial design and things like dose optimization, right? And one thing I will mention is I recently co-chaired a working group with the WHO on the regulation of AI for health. And I think this took 3 years. Okay, so it started during COVID, and by nature of being a co-chair, I eventually was speaking to the FDA every week, right, over Zoom, speaking to the EMA, MHRA, HSA here, a number of ministries across the world, including HBS, people from the Lancet. And I think the challenge with AI right now is that our discussions largely revolve around the same script. Okay, we go to an event, we talk about AI, there's discussion of data. You need good data to implement models, right? You need garbage in, garbage out, right? It's the same discussion over and over again. And if you even look at the publications that are looking at AI, I call it a lot of prediction without implementation, right? Where we take, everybody has the model that they like, they take data from various sources, they have internal validation, external validation, but the real test is when a clinician makes a decision prospectively off that model. And we are very much lacking in that right now. So prediction without implementation at the end of the day is an academic exercise. All right. And if we think about drug discovery and development, I went on a search to look for user adoptability and perception of AI discovered therapies in the clinic. And right now, there hasn't been enough yet. I think there's going to be more opportunities for that. But doing that engagement now matters, right? Because in the end, there have been drugs discovered and developed without AI that are the only drugs on the planet, okay, that exist for certain indications, particularly for antimicrobial resistance. And one I teach every year in a use case study or a case study analysis is Achaogen's drug to address carbapenem-resistant bacteria. At one point, basically the only drug available, right? And it still went out of business, right? Because in the end, clinicians, even though this drug was approved, even though it was WHO list of essential medicines, clinicians just didn't prescribe it, okay? And so in the end, it does matter, right? Beyond getting a drug to market, really looking at the entire workflow to ensure that AI discovered or not, these solutions are actually used. And so for this working group, it was very interesting to look at 3 years. In the beginning, that first call we had, I remember it vividly, it was exactly the script. Data should be unbiased, garbage in, garbage out, quality of the data, data sharing, data transparency, data security. And I was like, you know, we can't spend the next several years just putting out a document that just says that, right? Because there's going to be 1,000 other documents that say the same thing, right? So our document won't be very useful. And so as we evolved it, we started to show that not all AIs are the same, right? And if we have one document that gives that general narrative, it's going to completely miss an opportunity to say, look, there are different types of AI. There's different uses for the AI. There's different data requirements for the AI. Let's not blanket put everybody into the same umbrella. I'm not even going to go over this slide except to say that we run a bunch of trials, all right, using dosing to optimize patient treatment. But I'm going to run through one case, right? Amazing team here at NUS. And I'm going to show one piece of data. which is a patient right now who's still being treated for a very rare blood cancer here at NUS, at NUH. And the patient's had stable disease for, it'll end up being 3 years, right? Which I think beats historical benchmarks, ultra rare blood cancer. And one cool thing was eliminating the need for blood transfusions for this patient. And it was using a different approach. This patient was not getting this kind of population-derived dose. And the trial was actually supposed to be at most for like 18 months or something like that, right? And then no matter what, patients move on because they typically don't respond anymore. But the patient asked to stay on. We did an amendment. The clinician asked if the patient could stay on. And we can see how this patient was just evolving over time, right, with this cancer. And the thing is, this paper is in revision right now. And funny enough, the journal we're at doesn't take case reports, but this is clearly a case report, right? So oddly enough, it went to review. And one of the reviewers was like, you need to recruit more patients. Okay, get more patients. And I think the editor gave us 4 weeks to respond. So I think the editor basically was okay for us to blow off that comment, right? And we were like, this is a rare disease among rare diseases, right? And so it's not so common. It's not a mouse, right? I can't go and look for 10 more mice. It's a human. And it's fairly evident from the reviews we're getting for this and all the other ones we're doing that it's very rare right now to see uses of data and AI for prospective and interventional uses, which is why this is an important workshop, because we're talking about drugs, we're talking about therapeutics, right? We're running a bunch of other trials, multiple myeloma with Weiju, immunotherapy is actually recruited with Rogoff, and then we have a larger trial for hypertension right now, right? I think there's a lot of important uses with small data. So I'm gonna start moving towards the end of this, okay? Something really important that I learned during my time speaking to regulators is, so about 2 weeks after the ASCO talk, I got contacted by FDA to come and give a talk to them on getting the dosage right? Because now for oncology, dose optimization is like a requirement. And this is something I learned. I had no idea, and this was not long ago, was if you look at convention, if you look at a regulatory perspective, the term optimization means escalation. Dose optimization is finding the highest dose you can give. That's what it means to a regulator. And what we're doing here, optimization means the best patient response. Those are two totally different asks. And so when we develop drugs here, with or without AI, I think it's important that we don't just default to how drugs are developed elsewhere in the world, and that we explore new trial designs for the drugs that we develop. And that's exactly what we talked about with our 2 respective FDA commissioners. All right, so the final set of slides, some challenges moving ahead. Not all AI is the same. I think it's really important we don't have discussions where we kind of just keep repeating the narrative until something changes, right? The use cases can be very diverse. Ed covered this, right? There's AI for drug discovery and development. There's AI for medical devices. There's AI for diagnostics, AI for operations. AI, very big now in generative AI, and then AI for intervention. These are all very different uses, and we need to look at them very specifically. The data requirements for each of these will be different. Small data, Ed talked about some of the stuff we do. Big data, sure. What about no data, right? When patients walk in the door and we're going to treat them, we have no data on them because nobody else's data will be used to treat this person. Right? How do we build that understanding into the regulation frameworks? And in the end, who are the users of the AI we're developing? It's not always the patient, okay? Or it may be the patient and many other people. When more and more drugs are developed with AI, in the end, the patient's going to get that drug, hopefully. But the doctor's going to need to be okay prescribing the drug. The insurer is going to have to be actually willing to pay for the drug. And we need to have those discussions early because implementation frameworks can be very different. I think my last slide, when it comes to AI for science, I think AI for all science, right? There's behavioral sciences, there's regulatory sciences, there's clinical trial frameworks that we should develop. Engaging regulators early matters. And we live in Singapore. Our regulators are like right there, right? Right there. We should be speaking to them often. And they're available often, which is a very unique advantage that we have. And assessing economics early matters too. So a lot of what you saw is backed by diverse organizations and collaborations with pharma. Always happy to keep in contact with everybody. Thanks.
Great. We'll save questions for the panel discussion at the end. So next up is Yueming, who is an expert on AI and medical imaging, and who can answer maybe some of Ashok's questions during my session.
Okay, thank you. Okay. Okay, thank you. Thanks for the introduction and also the invitation. So it is my great honor to be here to talk about my thoughts on this promising topic. So my research is mainly focused on the AI for medical image analysis. So developing various of AI techniques to analyze the image. So here, we already know that in the modern healthcare, medical image techniques plays an essential role. So like the CT, MRI, and also like the X-ray fundus image, which is the clinicians will require on this to do the disease diagnosis and assessment. And also in the intraoperative care, the surgical video captured by digital camera can provide the surgical activities and also the tissue functions to directly assist the surgeons to do the treatment. So we already know that this kind of data has already been exploding, and automatically analyze this data is very important to assist clinicians or surgeons to have a better diagnosis and treatment. So for now, we already know that AI giving by like the increasing computational resources, AI have been developed and have achieved a remarkable success in various of various of medical image analysis. And the AI can automatically interpret this data. So why automatically interpret the medical data can help the clinicians. So for example, as shown in the right corner. So AI can directly segment, for example, the brain tumor, some certain region, and AI can already help like the concerning areas in the image. For example, in the right corner, we see that AI can directly segment the danger zone or safe zone during the surgery and to help the surgeons to pay attention on this area. And so totally AI can help to improve the accuracy and efficiency in the disease diagnosis and assessment. And furthermore, if we equip these AI techniques with, for example, the augmented reality system, it can help us to build up the medical education and training system. And also AI can equip with robots. So it's basically the building block for next generation of intelligence autonomous robot. So we can see that the AI assistant currently can relieve the workload of the clinicians, and it can also reduce the learning curve if we can develop the intelligent medical education system. And eventually, it will avoid the long waiting time of the patients. So this is generally the workflow of the healthcare procedure. So currently, AI have already helped with the data management, data analysis, and what we want to do next is we want autonomous AI. AI can help autonomous diagnosis, and eventually we look forward to augmented hospital. So my recent principle exactly routing this promising direction. And I have developed various of network architecture and learning strategy. And so in today's talk, I will focus on two topics. So the first one is foundation model, very hot topic recently. And also the foundation model with zero-shot learning. Zero-shot learning means no data required, no annotation required, very limited effort we need to do before we really use AI in the real world clinical practice. So this will dramatically reduce the resources cost to enable AI to have a real clinical practice. And another part is the multi-modality AI with the audio, with the test, and also how we utilize the AI, generative AI to 3D metaverse. So this can provide the interactive experience with the patients, with the clinicians. So it will provide a personalized and immersive experience for presence medicine. So the first part is foundation model, the vision foundation model. You may be quite familiar with ChatGPT, right? So this is actually called large language foundation model. And we also have a vision foundation model, which called SAM, segment anything, which is proposed by Meta AI. So in this segment anything, it assumes that it only requires the users to give a prompt. So here prompt may be a point, a click, or the bounding box, where the users wants the AI models to directly segment. And the segment anything can do like for any image from the natural domain. And segment anything model, only this one foundation model, they can directly segment the current region point by the user. So this is quite promising because when we have another image, for example, another image from different scenarios, this kind of AI models can directly output without retrain. We do not need to collect the data, annotate the data, and we can directly use this segmentation model on this unseen task. So we already require these capabilities in medical domain. So as I mentioned, so we have various medical modalities like CT, MRI, X-ray, also like the pathology image, like the genome, or different kind of modalities. So in original AI pipelines, when we have a new task, we want to use AI to directly solve it, we need to first collect the data and then do the data analysis, then do the data annotation. And we need to use the AI, build up the AI models to train it. So it will actually cost a lot. And in the real world clinical practice, as mentioned by Professor Ding Ho, we really cannot have a new users and we train a model, we collect the data. It's actually not that feasible. So if we can utilize, if we can build up our own medical foundation model, basically it can tackle this kind of problem. So what we first do is we test segment anything performance in medical domain. So segment anything, they means that they can segment anything, right? But actually in medical domain, it's not that unsatisfactory. Basically compared with the original AI method, with the results shown in the blue, this blue bar, compared with this, you can see that segment anything model cannot achieve quite good performance. But we can definitely use this segment anything model because this model has a lot of knowledge learned on a large scale natural data set. So how about we utilize this to benefit medical tasks? In this case, we can only use limited data and train very limited parameters. So it can already reduce a lot of cost. So here we build up our medical SAM adapter, which is the first model to adapt a powerful segment anything model from the natural domain to medical domain. So we only require to fine-tune a small number of parameters, and it can achieve a high generalization on various of medical tasks, and we only using a very limited data to retrain it. So here I show the results. So our method achieve a state-of-the-art performance over like 19 medical tasks and on various medical modality. For example, the CT, you can see the results, the multi-organ segmentation from CT, and also for the surgical video, we can do the scene parsing directly. And from the fundus image and also MRI and also some ultrasound image. So we can do a lot of tasks on a lot of medical modalities. The second part is that, so we know that the MathStem adapter, the first model we developed, we still require a small model parameters to update. And for each task, we still need limited data to fine tune. So is this possible to build only one foundation model with high generalization capability, and it can tackle various medical tasks without need of the training data and model update? So which I means zero shot. So here we built up this model and we can really achieve it. So we call it one prompt. One prompt means that after we train the model, when we receive an unseen task, we only require the users to give a prompt. So the prompt is just same in the prompt of the SAM. So it can be a click, and it can be a line, it can be a like bounding box. So which the users want our AI models to segment. So our models can directly apply on these new tasks without any retraining. So this is our overall pipeline. I just give the technical details here. And so these models can achieve promising results on over 14 handout data set. So this data set actually did not seen in the training process. So this is totally new data. And our model do not need to retrain. We can only require the users to give a prompt, and our method can directly output the segmentation mask. So you can see compared with the ground truth, our method can achieve quite close results with the ground truth. So it's quite promising. And the next part is about how we interact the image with another modality. So in this case, I show the test or the audio. So for example, there are a lot of applications. For example, automatic generates the radiology report and also do a video question answering, for example, in the surgery treatment. So in this case, the surgeons or the patients can directly interact with the AI assistant. They do not need to wait the senior surgeons because generally they do not have too much time to answer every question. So our AI models can directly help this. So this is also very useful. And we can develop the multimodality foundation model, which means the test and image modality foundation models to improve this. So this is our overall pipeline, the AI pipeline, and we can also achieve a quite good performance. So the left corner, we show the results when we're giving an image, the current situation, and also the question. The AI models can directly output the answer. And the last part, I would like to show my research work is about how we implement this foundation model into the metaverse, the 3D environment. So it can directly help the navigation and also the medical education. And we utilize the very novel Gaussian splitting techniques to do this. So this is also interesting. And this is the results. So we build up the whole, we build up the surgical environments and render it in real time. So this is the general overall pipeline, I think, which will be the future promise. So currently, we are at the vision foundation model stage. So compared with the traditional AI model, we need to design the AI models for the specific task, and it's actually not that feasible and it's very, very expensive when we utilize it in the real world clinical practice. So I think vision foundation model is a future promise, and we already did very preliminary works on this. And the next step should be like the multimodality foundation model. How we incorporate the how about we incorporate the image with another modality, like the test audio genome and also like EHR, something about that. So after that, we can utilize this kind of modality. And during the foundation model build-up, we can also utilize other learning techniques to achieve a higher level capability. For example, zero shot, continue learning to tackle the task heterogeneity, and missing modality foundry learning to leverage the multi-center data. And we can also incorporate this foundation model capability with another, for example, augmented reality system to build up mentors to achieve an intelligent medical training system. And also we can incorporate this robot to achieve the autonomous surgery. And this is what I think we can do in Singapore. So after consider the strength in Singapore. So what I think is that because we have various of distinguished researchers about biology, like you guys in MRI or mRNA or DNA, so we can develop our own multimodality foundation models. So currently we are at stage of utilizing language as image. But if we can incorporate other modality, for example, genome and EHR, it will be great. So we can, what I think is, this is a very good promising topic. And another is about generative AI. So how about we use the generative AI for medical training? Because this is actually the interdisciplinary research, and in Singapore, we have the expert in all related areas. And also the next, the final topics about clinical validation and real translation of AI. Because in Singapore, we have the leading clinical center, and we also have a distinguished clinical expert, and we always have a close collaborations between our engineers' wisdom. So I think these three topics are very promising, and we can do in Singapore. Okay, so this is my presentation. Thank you.
Great. So unfortunately, the final speaker, Nyam, is not able to attend. So we'll move directly into the panel discussions. But I did want to make one comment about Nyam's supposed, or what I thought his talk would be about, which is an area I actually didn't go into much detail in my original overview in AI and healthcare, is this idea of, and I think it was briefly mentioned by both Yueming and Dean, this idea of also AI in terms of database management, and how do we use AI to take health information from the patients as well as all the other data sets and analytics that various different groups are developing. And ultimately, how do we use that and manage that in the infrastructure? And then I know Nyam has been working really hard with the EPIC platform to develop the sandbox that I was hoping in my third and final point that he would, the sandbox that many of the researchers and people here can tap onto. So that's, you can always reach out to him separately for that. But how about we get a couple more chairs, and then we'll open up to questions. If people have specific questions, there's a mic in the middle that you can just walk up to. You do have to ask your question in the mic, or else it won't be recorded, unfortunately. Maybe Ashok. Yeah.
So thanks for to all of you for a really fascinating session that I think took us in a different and arguably even more important direction than the sessions in the morning, which is how do we take these developments and actually deliver them in the realm of healthcare? So my question is quite simple. We've seen in your different talks research and opportunities in the application of AI, all the way from new diagnostic or therapeutic indicating systems, suggesting systems like Ed's work on QPOP, N=1 clinical trials from Dean's work, and then systems which, for example, incorporate new ways of segmentation to be able to diagnose, or primarily in diagnosis from medical imaging from Yueming. In each of your areas, what do you think is the threshold for clinical validation so that the AIs that you are proposing or using are actually adopted in the clinic?
Yeah, so great question. Since I'm holding the mic, I'll go first. And so because we've gone through this journey with the spin-off company out of NUS, we've had actually a lot of discussions on this with both clinicians as well as regulators. And I think that at the very minimum, there's a combination of, for the clinicians, what they want to see is clear clinical trials showing clinical benefit. So on the clinical side, the clinicians want to be able to see in a clinical setting, does it improve patient outcomes? That's the minimum. And then also for insurance and reimbursement, that's the minimum requirement. For the regulators, actually the hurdle is lower. For the regulators, they first ask, well, what is your intended use? What do you plan to market? And then do you really actually achieve what that market? But I think ultimately at the end of the day, you have to show clinical trials where there's a clear clinical benefit above what is currently standard of care.
And thanks for that. But I perhaps will challenge you and Dean, because intrinsically, the types of systems that you guys are developing are n equals 1. So you can only validate per patient in a way. So what is the threshold? What is the barrier here? Dean alluded to that, but I think this is an important point because a lot of AI, I think, is moving in this direction, n equals 1.
Yeah, it's an important question. And so when Rob Califf was here, so he's the current FDA commissioner, we had a discussion exclusively on this N of 1 regulation concept, which we learned N of 1 means different things, especially for us versus what the FDA views N of 1. So FDA, when we first mentioned it, we had to kind of clear that all up. From the FDA perspective, the N of 1 is like a gene therapy, right? Designed for a specific person. You give it to that person. There's really no other way to validate off of anybody else. And then when we said, well, no, we're actually using a patient's own data to manage only their own care, that still defaulted back into gene therapy. Like, yeah, it's their own data to design their own therapy to manage their own care. And so we kind of spent the whole time reframing what a trial looks like, where it's N of 1, say, regimen design or N of 1 dosing, but we have pre-existing data from the individual on what they failed. So you get positive predictivity, negative predictivity. In order to make the dosing profile, we need to see the patient see doses that are the standard doses, and then they embark on this treatment. And so there are some elements of patient as their own control. There's positive, negative predictivity. But so when we explain that our N of 1 is each person's being managed N of 1, but it's actually a cohort, right? And so the studies, one I didn't go over was a cohort of N of 1 patients. And we actually spoke to HSA about this as well, which is for that scenario, we would actually rethink the statistics where we would look at variance between cohorts, where say a dose optimized cohort versus a standard of care cohort, the variance, intra-patient variance, inter-patient variance should be tighter. Even with smaller numbers of patients, you can actually get p-values that are defensible in this case. In our very first paper on transplant, we used that as an example for our regulator meetings, and they were very open to it. We're not there yet. I think the next step is and this is what we spoke about with FDA, which was let's actually do some of these designs, even in animals, okay? Even in animals, to show that these designs are feasible for human study. And the true feasibility is if a regulator will allow for claims to be made off of what are actually less patients than we think we need, but with outcomes that are statistically defensible. And I think the immediate first step of progress there was, I think the regulators would want to see claims that are performance claims first, and then eventually progress to superiority claims in terms of better overall survival, progression-free survival, et cetera. So I think right now, how we define N of 1, how we do N of 1 for multiple people at cohort level, I think we're there. I think now it's getting the statistical analysis clearly viewed by the regulators, and then the first performance claims to be made. And I think it's going to happen. I think that now having talked to 2 regulators, the heads of FDA about this, I think that it's all about now getting the trials done and getting the files submitted. But I think that beyond 1 case or 2 cases of this happening, there needs to be regulatory agencies teaming up for this, because the final thing I'll say is Right now, when a trial is designed for drugs, people are randomized to get this dose, they're randomized to get this dose, or they're randomized to get that dose. And if a person comes in and gets randomized to the max dose and they don't respond, they are kicked out of the trial.
Yeah.
They're non-responders. You'll never have known if they could have responded to a different dose. And so what do? You recruit a ton of patients, right, to have the power to give you an answer that is defensible. The question now is, can we learn a little bit more from each person to need less people to determine if a drug in fact works or not? The final thing, though, is if we are saying, hey, dose optimization is cool, right? You use our approach. The end game, though, is do you have to change the drug label, okay, in order to action what we do as a commonly used platform? And fortunately, when we spoke to FDA, they looked at this and they said, as long as the doses you are giving are allowable doses and the label allows for those doses to be given, you actually don't have to change the label. You would simply regulate this software as medical devices. So we actually got that answer out, which was critical because, you know, we're engineers. Every idea we come up with is the greatest idea known to mankind, right? But the reality is, if the regulators are like, yeah, you have to change the label of every drug that you want to dose optimize for, it's a black hole at this point. We shouldn't even bother. And I'm sure Yuan would have ended our project before it even started, right? So I think ideas can be great and we should keep pushing those boundaries. But in the end, that regulator sitting saying, you don't have to change the label based on what you're doing is an answer we shouldn't wait 10 years to try to get.
Sure.
Right? We should try to get those ASAP.
Thanks, Dean. I'd also be interested in hearing Yueming, because I think you have an opposite problem. Numbers are not the issue, but it's a way, when will pathologists be convinced that your software is good enough to do them out of a job? And when will radiologists agree to be replaced by your algorithms?
Yes, thank you. So actually in AI, there have a technique called prompt engineering. So like the work I present about one prompt. So basically we can ask the users, for example, ask the different patients to give their own idea that we call it prompt. So this information can be incorporated in our AI models so that the AI models can tackle various modalities and various situations. So this is the core technique.
But what I'm asking is, when do you think your algorithms will be clinically acceptable? Okay. Because you can develop all the algorithms, but as Dean has said, unless doctors adopt them in the clinic, unless they say this is valid, it's not going to happen. So I'm asking, how will you reach that point?
Yeah. So basically, when we design a new AI study, we will first talk with clinicians to see, like, is this task we are going to solve is clinical valuable? So for example, like the segmentation task, the one prompt segmentation task, the prompt tab is what we discuss, the results we discuss with clinicians. So we will like utilize the prompt which is suitable or comfortable for the clinicians and patient. And also actually have done a lot of collaboration projects with clinicians. And we will validate our AI models with two types of results. So the first one is like the clinicians directly do the process of the image. And another is the clinicians will, based on the AI predicts to do the analysis. And we found the results that, so from both accuracy and efficiency, it can improve a lot after the clinicians based on the AI prediction. So
do you have plans for regulatory validation, not just at the level of clinicians? Because otherwise you'll only influence a small group. What are your plans for regulatory validation?
Yes, this is very important question and also a very, very interesting question. So yeah, we do have this. So because after that, we can really apply the AI method into the real world like practice. So what we want to do, we need to first to do a various of clinical trials. So currently we also have our system developed, software developed, and we hope that the clinicians can utilize this and give us our real feedback. And we already connect with clinicians and to do this.
Thank you.
Can I jump in really fast? Yeah, really important question about will doctors actually use it? I think that we often think of the user as the patient only in the end, right? But I think there needs to be this discussion of evidence and behavior change. Because as we've seen, as I mentioned, there have been drugs with clear evidence that they work, but the doctors don't prescribe the drugs. And so I think that implementation is as much evidence, is as much economics, and is as much behavior change as anything. Because in the end, for true practice changing workflows, there are many components of that workflow that will need behavior change, including the doctors, the nurses, the pharmacists, allied health, caregivers, and then patient acceptability. And so I think that, interestingly, you know, at Wisdom, we just onboarded a behavioral economist. And when we started Wisdom, I never imagined that we would have at one point 7 behavioral scientists and now a behavioral economist to really get at the heart of how do you change behavior of everybody in that workflow. And there are cases where the doctors will use it, but the economics of the device or the technology can't justify the actual reimbursement for it. And then that's where things fall apart. So I think we need to think about this.
What is the ethics of your
Yeah.
Thank you. So doctor can also make a mistake. They have some level of accuracy. So AI, you also have like a quantitative accuracy. I want to know when your AI model can reach to the accuracy of an average doctor. So that probably is the key point, which I think is what Ashok is asking. When, I mean, if you are higher than the doctor, average doctor, then your AI model really become useful.
Real doctor for the
No. So firstly, sorry for missing the morning, and I got here because I wanted to hear Niam, because I wanted to hear his experience with everything that he's been doing. But unfortunately, he's not here. But even in the AI for Genomics workshop that Millie and I kind of did, right? One of the key themes that kept coming up repeatedly was the QC of the data. That was like, you know, we have all these ambitions, we have all these questions, the QC, how do we get all the different data sets? I think we're very lucky. We come back to the theme of these workshops. What is Singapore's advantage, right? One key advantage is we have pretty good EMR. All our EMRs are coming together through trust. I think that's great. But that's one side of the wheelhouse, right? So all your RNA, genomic data, imaging data right now, the harmonization of that, the QC of that, all of that still remains very disparate. And I think that was a theme that came out repeatedly in our thing, was how do we, like, if we really want to leverage all this, right? Like big challenges, right? I don't think anyone has really solved this. So my question, and also comes back to that, is until we can resolve this bit, then the confidence of the evidence and all that becomes then a problem, and etc. So I guess my question to you guys in the long-winded way is how, like, in this grand challenge that we're about to do, how do we ensure the cues, how do we pose challenges that will help us address this issue, fundamental issue of getting all these various data sets together in a way that FDA will agree that this is good enough, and etc. etc. I don't have answers. I'm just asking your thoughts.
So I think they're both somewhat related questions, and maybe I'll address Young's first, because that one's the easier one for me to do, because you know, we've been dealing with it, especially really quite deeply for the last year. And so I think a lot of these, especially in the diagnostics area, when we use AI in healthcare, specifically in diagnostics or prediction of treatment outcomes or prediction of disease state, this applies for medical imaging as well as our platform. Really, the critical criteria for clinical accuracy comes down to many of the established analyses that they already do for NGS panel testing, for other traditional diagnostic tests, which is basically like calculating the NPV, the negative predictive value, positive predictive value, total predictive value, specificity, sensitivity scores, the AUC of the ROC, And for many of these already with non-AI enabled diagnostics, there are already clear cutoffs for what is clinically acceptable in terms of moving forward and in terms of trying to commercialize. So generally for those, the baseline cutoff is like having an AUC or a general predictive like overall accuracy of above 70-75%. It's kind of like the baseline. So anything below that, you shouldn't really move forward, right? And ideally, even then, they want to see things above 80%, 90%. Obviously, 100% would be amazing, right? But generally, a good diagnostics test is generally you're starting to get good diagnostic tests once you start getting above the 80% range. So for instance, with like GastroClear 1.0 was, I believe in like, I think I presented like 73% or something like that. But now, even with the new modified data and the new analysis, they're now moving into GastroClear 2.0, which now is above the 80% mark. And that's the one that they're really trying to implement now with the EMR, with the Epic, and how do they get it into the electronic database. And then I guess maybe on that, to move forward then in terms of how do you QC these beyond just clinical accuracy, this is something that regulators are struggling with. And ultimately, they're basically trying to like implement certain specific protocols, certain ISO certifications, in terms of the workflow of how you collect the data, how do you store the data, how do you record the data, and there's these quality management record systems that essentially all these groups ultimately will have to have in place, these QMRSs to ensure the quality of the data that's coming in and how that data is handled, how the data is stored, how the data is processed. And I think for that, the key is to align, as long as the groups can generally align on these QMRSs, then that's one way that we can standardize and regulate. And obviously, this is an emerging area that the FDA, the HSA, and the MOH are all kind of working towards, but it's still an evolving field in terms of determining what are the minimum thresholds and what are really those SOPs that what are the standards really for those QMRSs that ultimately all groups will have to implement it for their QC analysis.
So I was going to really quick. Yang asked a very good, important question about whether we're out competing the doctors, because I think for adoption, it's important to have a benchmark of where are we actually. A critical question. So I'll get specific, right? For transplant, we are discharging our patients about a month earlier compared to clinician-guided care. And that's in the US, right? So for those who don't know the US economics of transplant, UCLA is about $30,000 USD a day for an ICU, right? So we're getting people out about $1 million reduction. But for 30K, you don't even get lunch, by the way. Okay, it's just for the room, all right? So in terms of the economics, in terms of and this is interventional. This is not a prediction off of data. This is doctors treating with us and doctors treating off of standard care. So there's an economic and an efficacy-driven outperformance there. So that's clear. For oncology here, the current clinician acceptance rate of the dosage recommendation of cure rate is about 98%. And the bulk of those other 2% is because the patient got COVID or something. So the doctor just has to go off this treatment protocol and just hold treatment or give a max dose or something like that. In terms of the overall average dose given to patient, we're seeing about a 20% reduction in the dosage needed by the patient compared to what they would have been given. And we haven't done any analysis yet on what has that resulted in less readmission due to toxicity, et cetera. And so I think that, and then it's such an important point in that I actually don't think even accuracy is enough to say that the AI should replace or should be used exclusively, because in the end, I think that the actual implementation and to do that full switch, I don't know if saying, look, if you use the AI, your patient may go home 10 days earlier. That alone will not necessarily get a clinician to just wholesale use the approach. And I wouldn't blame the clinician for that, because when we interact with them, it's, experience matters. There's a lot of other factors that go into the real implementation, right? And so, but at the heart of it, there has to be some numerical benchmarking. And I think we're getting there. One thing and we get, the regulators do ask us, and we ask the regulators back, like, what type of variance p-values are you looking for? And we're having those discussions already. So I think we're doing okay. But then the next part is there's still so much more to address after that. And one thing about the data, I think that something that's raised is, I worked on trust as well. I think that when we talk about data, RNA, all the imaging data that's there, I will argue that the data, from an interventional perspective, I'll argue the data we need to actually truly optimize intervention actually doesn't exist. Okay, we don't have that data. We don't know, no database in the world actually exists for that. And the reason is, when we're going to treat a patient, whether it's with us or with anything, or we're going to just go mine clinical trial data, the simple question we have for the doctors is, do you have patients who got this dose, patients and got this dose and got that dose? And 100% of the time, the clinicians will say, or the company will say, yes, we have that data, tons of the data. And I'm like, let me clarify. I need each patient to have seen this dose, and that dose. Not 50 guys that saw this, 50 that saw this, and 50. Oh, actually, no, we don't have that data. All right, because patients evolve over time, and variable response as a function of variable intervention, that data is not there. Okay, that we know. And that's why we have to prospectively collect that data. Right. And I'll be fair. I don't think we should re-engineer clinical protocols to start doing this for everybody until there's some evidence that that type of data collection is useful. Right. But so far, every study we've run, whether it's hypertension, oncology, both blood and solid, we clearly see variable response as a function of variable intervention that's dynamic within the same patient. I want to be very clear. Our goal is not to low dose cancer patients. That's not the goal. The goal is to find more responders. And every study we do, we find people that don't respond at the population-derived dose, but can respond at a different dose, which tells us we're missing responders. That's the ask we are making of ourselves. It's not to come in and ask clinicians, just low dose everybody. Let's redefine the population dose to be lower. That's actually not the ask, right? Because I think that's not accurate. I don't think that's what we should do. I think that we would never ask the clinicians to do that. And I think we need to be clear with what we are wanting from the AI approach.
So,
Okay. Actually, you probably partly answer my question, but I still want to know. Certainly, AI model is not perfect or it's not good enough. So, what is the key challenge or bottleneck in your viewpoint, or each of you guys in your field? Because this is also important for us. This white paper or like this study report.
I can answer this question. So the very first thing is about the accuracy of AI model. So currently what we want to is to not like replace the clinicians and surgeons with AI. We want to develop the AI assistant, which can assist the clinicians to increase the accuracy and efficiency during the diagnosis or treatment. So basically we can do like two experiments. So the first one is the diagnosis or treatment only by clinician. And the second one is utilize AI based on the AI predictions and to further like refine or among the prediction. So we basically do various experiments on that, and we can see that AI can help to further improve the accuracy. So this is what basically we want to develop. We want to develop this kind of system to assist the clinician. And the current, the main challenge, I think, of the AI model is that because we have various of tasks and also various of medical modalities. And we also have a diverse within the patient. So basically, if we developed, so basically in the original AI model development, we need to collect the data and do the data annotation and then train the AI models for this specific task. But it's actually not that feasible when we really implement in the hospital because it's too expensive. So what we want to do is to build up a foundation model, as I mentioned. So this only one models can handle various of situation, various modality and tasks, and this can also cover the diversity of patients. Yeah.
I don't think we should stop the vision at citizenship, but really,
Okay.
I think we should be thinking about
Replace the clinician, right? Yeah.
What does SMC have to say?
You know, doesn't matter, right? But I think that we all. So it's fine, right? To try to replace it, but the problem is right now, anything about you can't replace, you can't replicate it tomorrow in the next system because the way anything is done is not the same. So in terms of how data is labeled and whatever, and use the same touching machine, do the same imaging. Tomorrow, if I say from Singapore to NUH to deploy it right now, right? We can't because the data formats are different, you know, so we need, I mean, I think clinicians don't mind being replaced, but I think we need to be replaced with something that can then be, a doctor with the same training can move from hospital to hospital, right, today, right? I can go from SGH, I can go to Changi, same algorithms in the brain works. But the algorithms right now that AI is developing is so, in some ways, very specific to that environment that's being done.
Yes,
so that's
exactly
a
what
challenge. So I you know, in terms of speaking of what are the grand challenges, this is one of the challenges. How do we make it such that it's more easily replicable?
Yes.
Right.
Yes, exactly. That is exactly what I mean.
I want a solution.
Thank you so much. Yeah, and also about replace the clinicians. Yes, that's definitely what we want to do. So currently, the AI models can help to help the surgeons to do some simple but tedious tasks. So, for example, generally, the patients will have a talk with the clinicians to ask the situation. So this kind of very simple or tedious tasks, we can utilize the AI models to that. So for example, the chat box, and also based on the ChatGPT, we can.
So in a simple word, it's just the quantity of the data.
Yeah, exactly. Not that. And also another point is that we can develop the training system. So what we want to do is that we can train the clinicians from 0 to 1. So in that scenario, for example, a very training or very junior clinicians, basically the AI models can teach something within this. And this is what we want. For example, we can incorporate the AI assistant in the training process of the surgeons. So when the real clinical practice, the surgeons are already familiar with the AI system, so that it can utilize this more frequent when do the diagnosis. So this is another procedures we want AI to do to effect the clinical practice. Right.
I just wanted to raise one quasi-philosophical point, actually, because I think now we're talking about what is the threshold at which AI-based algorithms become useful in diagnosis and therapy management. So I think everyone in this room, including the clinicians, knows that some people, some doctors, are better at diagnosing and better at treating patients than others. In a way, you could consider those doctors, each individual doctor, to represent an AI algorithm. They are processing data, they're using their brains, they're based on their own data from their experience, and they are outputting. And some of those outputs are clearly better than others. That's why some doctors are more popular than other doctors. So we're now arriving at a point when we are talking about AI algorithms meeting a basic standard as Ed said, which is, say, 80% accuracy or whatever it takes. Are we, by implementing this and potentially replacing superior RIs, I call them real intelligences as opposed to artificial intelligences, actually impeding the benefit of patients? I know it's a quasi-philosophical question, but as we get closer to this point, we're not talking anymore about RNA molecules or drug development. We're talking about real people. So this is an issue.
Yeah, that's a great question. I actually think it would impede if we were wholesale just replacing. Sorry, maybe some need to be replaced, maybe some don't. I think wholesale replacement, and that's actually why when we all of our workflows, we actually have the user engagement, the qualitative study with the clinical community, and that's everybody, to actually find out, to ensure that how we integrate this in is not a black box. Okay, so in the end, the clinician themselves build the small data set prospectively that they themselves will approve at the end for the patient. So they actually see the whole journey all the way through. It's always a clinician-driven approval at the very last step. There can be a whole separate question of did the really good person, did the optimal RI override the AI, and they were wrong? Who knows? And that one, we'll have to kind of look at population data over time to see. But I think wholesale replacement will impede. Back to Yang's question about bottlenecks, an important one, I think, for everybody is, I still think AI is being viewed as a fairly umbrella platform. And I think viewed as an umbrella platform, we're all being subjected to the same criteria, right? The data sets, the quality of the data. The thing is, as much as we talk about the quality of the data, for some people like Ed and I, that actually is an irrelevant criteria for us because we don't use any of that data in there. But it's very important for other people. It's critical, in fact. And even though we know that AI has different use cases, it's still being lumped into the same category, right? Because the same pillars are being asked of those doing diagnostics, medical devices, intervention, drug discovery. It's the same kind of checklist that's being raised. And then in the end, how we regulate, I think we even went through that, right? We were asked about external validation sets for our work, but it's actually a completely prospectively validated use of the approach. And we're not merely trying to validate across different sets of data to see if it all lines up well, right? So I think the one major bottleneck is, and it's going to take time, right? But AI, like many other fields, everybody coming in with a different way to use it, a different type of the AI or technology in general, is being subjected to the exact same set of questions, the exact expectations, an assumption that even if we align data across every healthcare system in the world or in the US alone, that suddenly we're all going to be helped by this, that won't be the case. And I think there should be focus groups and working groups that, frankly speaking, of course, Yueming and I, we collaborate, but there are supposed to be regulatory working groups, clinical validation working groups, workflow development, where actually we should be in completely different groups, right? So that we can iron those out first and then come together to, or do both in parallel. Workflow integration, but domain-specific sets of regulatory validation, what data even means. Right now, we're still in this very generalized approach. Yes.
So actually, maybe.
The mic, please.
Yeah. So while you're going to the mic, maybe I'll answer Ashok's philosophical question about the clinicians. And this is actually very interesting, talking to a number of clinicians with our work, is that you do find very similar to ML versus AI, that different clinicians fall into different, along a spectrum, right? Some clinicians, Joanne, I know you're maybe on the more generative, more generalizable AI side, but just like there are different models of AI, some clinicians, for good or bad, generally fall in the very clear, like there's a specific set of rules that within this algorithm, as long as these rules are followed, then I'll move down the decision tree. anything, any query that's asked outside of that cannot be answered by the decision tree and the rules set up within this specific algorithm, I can't answer that, and I won't be able to answer it, and I won't move. So then when they get a patient or a clinical problem that's not within the defined rules and decision tree of their algorithm, they can't help, right? On the other full end of the spectrum are clinicians that maybe represent generative AI, generalizable AI, maybe foundation models, these zero-shot foundation models that are being developed. And for them, then it becomes, no matter what the query is, no matter what the problem is, there's at least a framework in which to potentially create a generative hypothesis or a generative approach. It may be good or bad depending on how much data has been inputted into those specific areas, but they have enough broad deviations from those rule sets to allow them to kind of come up. And obviously, I think very similar to AI, you have to have different buckets for those clinicians. And I think the idea is with all the different modalities and the different AIs, how do we use, I guess, AI in data management, AI in infrastructure to collate all that data in a meaningful way that allows even the decision tree, rules-based clinician on this side get some valuable information from AI. Yeah.
And we only have 5 minutes.
Oh yeah. So maybe one final question.
Thank you. Regarding to the accuracy, I think we learned from the success story. As I mentioned, Yang mentioned, the AlphaFold, right? The structure prediction from sequence to structure. In fact, it's not success in one shot. It's nearly 15 years, the computation, the CASP. Every 2 years, we have submit, the known sequence, but already known the structure, but we keep the answer. Then competition, all the group in the world submit their prediction. Then we do the very objective, assessment. Then we see every group, you do not need to say how you are very good, but then we do the objectively assessment. Then it's very clear which method, at which level, and what is the gap. So very clear. I think similar. If we can similar at the clinical level, do we have such objective competition? Means that every model we can come up, but then we get a certain objective benchmark to let every model to check. Then we year by year we know what the gap, what part we should implement.
So I think clinical accuracy is one aspect, and I think that's one area that, I think as we mentioned, there are clearly defined in terms of how can we outperform prior tests or prior platforms or prior approaches or current clinical practices. But I think for clinical implementation, it's a little more tougher than just accuracy, right? As we mentioned, especially with increasing complexity, there's also cost. This is something that we've seen. No matter how great it is, if the patient can't pay for it, or if the Singapore government's not willing to pay for it, or your private insurance is not willing to pay for it, it's not going to be clinically implemented, no matter how accurate it is, right? There's a number of other factors in terms of, things like time to decision. How does it add 2 hours to the clinician's time per patient, or does it reduce time per patient, clinician's time per patient by 5-10 minutes, right? If it reduces how much time the clinician is spending on a patient, then it might move up, even if it has potentially a slightly less accuracy than another platform, which increases a clinician's need to spend an extra hour on that patient, right? So there's a number of other factors beyond clinical accuracy, I think, that makes AI implementation in healthcare much more complex than just, can we set these benchmarks and every year set models head to head, and whichever one's accurate, let's move forward with that one, which is a little more complex than, drug discovery prediction.
Just one really short point to tag on. I think it would be really cool if there was some sort of global benchmarking for clinical algorithms. I think but then again, if we again do a benchmarking process, whether it's for protein folding, molecular design, dosing, diagnostics, et cetera, that's a tough ask for very different use cases. I would say we've got a clinician in the room, multiple clinically trained colleagues here. in the end, if we're going to do certain things in clinic, whether it's benchmarking or trial designs, trial implementation, to get to the heart of it, the patient has to say yes. Okay, if they're going to be enrolled in a trial and something's going to be tested on them, the patient has to say yes. And if it's going to be a trial for benchmarking, you're going to need anywhere from 500 to 1 million patients to say yes. The process of consenting a patient into a trial, depending on what the indication is, can be everything. It can be 50,000 different things. It can be exciting for some, it can be heartbreaking for others, it can be scary for some, etc. Right? So that step is to be accounted for. So that's why, again, I think we all agree accuracy matters. I think we agree that one thing we want to note is accuracy alone. I think there's other things we have to consider in that process. And so I think right now the bottleneck is how do we segment the asks that we have for each use case or each domain in a way that we can actually meet those benchmarks. First of all, find the benchmark, establish it, meet it in a time frame where the field can progress. I think all that's hard. I don't even have an answer to that.
Actually, what Yuguang mentioned about the CASP competition is the same. Even the protein structure prediction accuracy, what is the criterion? How to assess that? There's a long history, tens of years, people quarrel, and because there's many different ways. You can measure the global topology or clash or atomic level detail or function. So that is, the same thing. I mean, of course, this is, I agree, this is a cool idea. It's very important to push that field. Just like a CASP, push the strategy prediction field. This is dramatically beneficial. So if there's something set up in your field, certainly I agree there's a complexity, but it's still worthwhile.
Terrific. That is a fitting conclusion to a very exciting session. My thanks to all the three speakers and to all of the audience participants. Yang, it looks like you and Kedar have a real challenge for the future, which is how can you design an AI, not for RNA, not for DNA, not for digital pathology, but to change human behavior? That's the real challenge. Anyway, on that note, let me hand over to Yang for the concluding remarks.
Okay, it's not really a concluding remark. So I try to save you guys time, and I listen very carefully, and I never really go to a meeting listen so carefully because I learn a lot. And honestly, at the beginning I was worried about whether there's any discussion, because usually, if there's no discussion, then how can we go to this like a discussion panel? But fortunately, I see this discussion is almost endless. So that is a measurement how successful of this workshop is. Thank you for everybody, and also especially Ashok is a good organization. And I think this also the many of the questions discussion are very insightful. So different field have different criterion, and but I found really there's some things in common. And I try to summarize these three things that are in common. First of all this field or the question or AI development is data quantity is the first priority. Because just like in my example, for AlphaFold, and we have protein, millions of the protein, millions or billions of the protein sequences, and almost a million of the protein structures. That provide a really enrichment of the data set. Like the success of like a ChatGPT, everybody knows that. It's also because of the data set, like Wikipedia is so enriched. So that is success, very critical, factor. But somehow, like in the RNA structure prediction, it's not that successful. Partly it's because of the data set, or some of the you all mentioned in the precision medicine and healthcare. Data quantity is really a challenge, bottleneck. So secondly, you know, when I just compare the protein and the RNA, you know, also this the AI advancement compared with the traditional approach. The key point is the parameters. Parameter of this AI model basically is, you know, can be analyzed by the model metrics like embedding dimension. You know, if dimension is higher, you have more parameter, you can train better. But if you have more parameter, you need higher computing power, the GPU, higher level of GPU. So that is actually the major reason why most of the breakthrough in AI are achieved by this big AI company, like, you know, DeepMind or Meta and AI or this OpenAI and so on. In our field, we have actually many of the idea we already have. We tried that probably months or years before this, you know, DeepMind or others. But, you know, the challenge is the computing power. Of course, there's also the human resource is part of the issue. So that both of this, you know, computing and this data, which is NRF, really pay a lot of attention, recognize the importance. So I participated in several of the meetings, and also yesterday I meet with the NSCC. There is a substantial amount of this budget will be allocated for GPU project, specifically for AI for science. Because at the beginning, they try to put this into the NSCC. Later, I really insisted that we should have an isolated computing resource cluster, because AI for science, their data set, their structure are very different. So looks like they accepted that. So we will have like either NRF have a big cluster, you know, room here, or it's handled by NSCC, but it's we have our own staff, IT guys. So that is very good to address these two issues. The third one is that we know that given the data, you know, RNA data or the clinical data, you know, we cannot really change much, right? This is a life, and we have to deal with that. So the critical thing is that we have to have better or smart AI method. So actually, we have seen this all of this breakthrough of AI are all, you know, associated with some breakthrough of AI technology. Like, you know, machine learning has been there for many, many years. Why AI or deep learning just brought out like in this 10 or 20 years? It's because there's new methodology like a convolutional neural network. You know, they are really increasing, you know, vision and this image recognition power, like a ResNet, really increase the depths of the deep learning, make it a deeper quantity. Transformer make the new technology really make this large language model more stable and more accurate. So that is also the advantage or necessary of AI for science, because we will bring together or work together with this AI expert, and we propose our question to them, and then they will try to work together with us. And you know, they are really professional. So we have some smart students here, but we need to really respect those professionals of, you know, computer science and AI expert. So that is what I can say is very promising and necessary of this AI for science, which is supported by the NRF. So I'm very confident, and I also, you know, I spent a lot of time on these meetings and report, but I think it's worthwhile. So, but this is, you know, the meeting or discussion is very important for the white paper, which we're waiting for you guys' contribution. But there's still a lot to follow up. Like what I mentioned is like this white paper, and also the especially the May 10th, I hope you guys can come. This AI expert, the science of AI workshop should be very good for all of us if you're really interested in the AI technology. And also the concept of the paper or like a proposal for this support eventually will be, important for our effort. So there's a lot to follow up. So thank you again for all your attention.